let's start with Grace. Do you have any updates, Grace?
Oh yeah, so me and Eileen worked on the variant classification project a little. So I'll show our progress on the Excel sheet. So first we went through each of the genes listed on the bottom. It's basically the same as before. And instead of doing like the first 10 like most frequent variants, we went through and did everything. But one question I had for you, Dr. Che, when we were doing like the 10 for some of the genes like that couldn't fill 10, we did the ones with just like one, even if they didn't have a consensus across like more than one database. But I just wanted to ask, should we still do that now that we're having a lot more variants? Like, I mean, certainly for some of the ones with less variants, it does add to it. But I know we were talking before, like when we were doing Emma's project, for example, we usually did ones with more than one database for consensus.
Right.
The benefit to adding the ones with just one would be that, like, for example, this one, like mTOR and also like PIK3CA, you'll see that most of them are like oncogenic, except there's one that has like one benign, and, like one benign here. So yeah, when Dr. Eugen and I were doing like the calculations, like the ones that didn't have benign like ended up getting like some weird scores for some of the things we were calculating. So it helps to have both benign and pathogenic, but I don't know how like reliable this is.
And it was, let's say, for instance, that A8S was just described in OncoKB as benign and was never mentioned in three other.
Yeah, so CBioPortal shows like those 4 columns, and basically it was only flagged for that OncoKB column.
I think for entirety, the completeness, I think it makes more for the actual manuscript. It's more meaningful to have the full list, like not, having at least one annotation from any of 4. Does that increase your number a lot, or how does that impact your workload?
So basically I highlighted those ones in red. So you'll see it includes it like that much, that much, that much. It's not.
Yeah, it's not too much.
Addition.
Yeah. No, yeah, let's do it because some gene variants are going to be just one. Yeah, for completeness. Yeah.
Alrighty. So then, yeah, for BRCA1/2, they only have these. So
and I think for completeness, I think these tables, this is sort of like describing the workflow. Like this is the actual variant that you used. So I think these tables should all be supplemental tables for your actual publication. Yeah.
Okay. Would there be anything else I would need to include in this table then?
No, I think this is fine.
Okay.
Yeah.
That sounds good. Yeah, that's what the progress we made on variant classification for now. And so the next step would be, I think this is just something I will do to basically generate the codes for each of the variant to be plugged into each of the tools. And then like last time, I'll distribute it to like Eileen and Lena and myself to split up the work and run it through all the in silico tools.
Yep, we know the workflow, so. Anything else? Is that all?
Yeah, that's mostly it. I'm going to be working more on the poster for ASCO with Dr. Liam. I used the Northwestern template, and I just started copying and pasting stuff from the abstract. It's looking a little empty, but Dr. Liam and I are thinking of making like a table of sorts or like some sort of graphic to better represent the data than just text. But otherwise, that is it.
Great. Thank you, Grace. Since Minjung is here, Minjung, can you give us update?
I don't have much to give updates on. I just had a Zoom meeting with Dr. Shah and Dr. Army 2 days ago, and just talked about the next steps to take. So I think they'll be working on that. And for the tumor growth kinetics calculations, they weren't too confident about running the R script. So once they fill out the table in the right format, then I think it shouldn't take too much time. So they're going to fill out an Excel sheet for that. And they also mentioned that for the volumetric hyperprogressive disease patients, as of now, they're unidentified. And I think they told me that Dr. Eugene is trying to obtain the IDs of those patients. And that would be necessary in order for us to make a comparison between the volumetric versus RECIST-based hyperprogressive disease patients. So that we can work on later, but for now, they're reorganizing their data so that it could be run. That's all for me.
Great, thank you. So yeah, so we do have volumetric data. So work with Eugenio. Yeah, I think that's a big plus in our data set. Yeah, and this data, anything that comes out from this data can be oh, this is Peter. Emily's working from home to
Peter.
Peter, go to Peter.
Is there anything I can help you with in the meantime?
Always go to
Just go, just go to Peter. Yeah, so I guess Minjung, you know all the directions, you know what kind of comparison we need to make, all that. So something that you did in your meta-analysis, we'll do it with our own patient data. And I think although it's lung cancer, I think we can, yeah, did we discuss, so whatever you guys in this analysis discover, you can report it to, I think CISI. CISI would be good because it's about immunotherapy hyperprogression. And then I think the meta-analysis you submitted, I know you submitted the paper. Usually to get it published takes like 3-4 months easily. So one thing you can do is maybe you can submit to CITSI and then we'll look at, and then once the journal gets accepted, you can notify them this was submitted for abstract. And then it's kind of an embargo. You can publish it on the day when the abstract is released to public, something like that. Yeah, so I think you can still recycle it to CISI. The meta-analysis and whatever we get out of this analysis. Yeah. So let's shoot for that. Anything else? Any other insights? I think the big picture is that Dr. Yuri will look at all the radiomics features. And he's kind of doing, he's done that with hepatocellular carcinoma, and he's gonna do that with lung too. It's just taking time for him to build up algorithm and things like that. So yeah, that's in the pipeline. And the other thing is we'll send the whole DICOM file to Lunit. So Lunit will be also doing radiomics analysis. Yeah, and hopefully because they have the H&E slide, well, technology, problem is the paucity of the existing sample for that analysis, but you know, the goal is to really integrate pathology and radiomics together. Okay, great, thank you. All right, Junie, you give us update on what you guys have, where you are with the analysis or the data collection?
I'll be the one who updated, but give me a moment, I will share the screen. Because you're in the, wait a moment.
Maybe copy it.
Do you need more time? Should we get back to you?
Only thing that's gonna run us stairs. I got the forms here. We need to
5 minutes more after
yeah, actually.
Arnie, you're breaking. Can't hear you.
That too.
5 minutes, doc. You can back to me like after 5 minutes, please.
Okay, let's do Juhi. Juhi, are you ready?
Another question bank is a bit more.
I am like semi-ready, I guess. My presentation is not exactly complete, so I could present it today or I could do it next time, but whichever you prefer. It's not like 100% yet.
Okay. Do you want to present what you have now and see if you need to add anything? You can present the rest.
Sure.
Yeah, let's do that.
Oh. And they have it like you can go on Google, you'll see a lot of stuff for just like regular people.
How do I
But here, for medical.
transfer. Hmm. So basically, we were gonna look at the commercial landscape of blood-based cancer screening. And then we were gonna look at who the main players were in this aspect. So you can kind of divide it into multiple cancer detection and single cancer detection. So I'll start off with the multiple cancer detection. I guess everyone kind of is familiar with the Gallery test by Grail. It is the blood test that detects cfDNA through NGS, recognizing DNA methylation patterns associated with cancer. So it has 4 trials going on: the CCGA, Strive, Submit, and Pathfinder. So CCGA, they have results. They basically analyze blood and tumor tissue samples from around 15,000 individuals from 142 sites. Um, and more than 50 different cancer types were included. Uh, and they had like sub studies. Their preliminary results showed that the test was able to detect 12 kinds of cancer, anorectal, colorectal, esophageal, and so on. Specificity was 99.3, which is pretty high. Tissue of origin was correctly identified with 93% accuracy, and with the test having like the 67.3 detection rate for the 12 specified uh, overall, and like different detection rates for each stage. Lowest for stage 1. And the overall detection rate for all type of cancer was 43.9. Um, and Strive is another trial that they're doing, still in progress. So this one is directed to breast cancer for the most part. Um, and it's a study with like 100,000 women across 35 sites. Summit is a similar study, but this one is directed towards lung cancer in the United Kingdom. Um, and they are like split and they're stratified into groups depending on the risk of lung cancer, like smoking history and so forth. And they're also doing Pathfinder. Pathfinder had a preliminary result published. Pathfinder is not directed towards the single cancer, it's the multiple cancer test. They have around 6,000 participants, and they have 2 cohorts. One cohort is those with elevated risk, and the other one is with non-elevated risk. And they their interim result was that their PPV was 45%. So Gallery test is already out in the market for about $900, or like more closer to $1,000. And yeah, they're still running some of their trials. Cancer Seek is another test out there. It detects cfDNA and identifies A protein markers. And it also looks at the mutations and genomic positions. They did a trial with approximately like 1,000 patients, and the specificity was according to them, it was over 99% and 8 cancer types. PanSeer is another one that detects DNA methylation patterns linked to gene silencing, which may contribute to cancer development. And their study was a longitudinal study from the TIZO study. So they basically look at tumor DNA methylation. They had 1,200,000 healthy subjects to start off with, and then they followed them up for like 4 years. And then they had 605 asymptomatic people who later were diagnosed, of which 191 were diagnosed with cancer. They These are the results they reported. The 5 common cancer, and recognizing them was a specificity of 96%. It detects cancer in 95 of asymptomatic individuals who are later diagnosed. Another test out there is Pantone Detect. This one uses epitome detection and monocyte-based technology. They basically look at two biomarkers, the APO10 and TKTL1 in monocytes that basically ate up tumor cells and migrated to the blood. The results shown by Grimm in 2013 was they dealt with various cancer patients, and their results were sensitivity of 95.8% with a specificity of 97.3, and an FPR of 4.0. And a 2020 article showed that both biomarkers were positive in 100% of like 62 cancer types. EDGC is another company out there. They look at acquired mutations and cell-free DNA in blood by NGS. And they screen for 10 major cancers. And they were going to offer a service called OncoCatch. So I guess they want to be out in the market in the first half of 2023. They reported their sensitivity to be 93%. I couldn't exactly find their like article, so I'm not exactly sure like of the scientific article that reports the sensitivity.
So what about specificity?
They didn't report it in that particular article that I read. And I couldn't find a separate published article, so this was like one of the things I wanted to update. So a lot of the studies deal with like multiple cancers, but then like one of the review papers talked about how one size may not fit all. And then we move on to like single cancer detection.
Can you go back to that slide? I just wanna,
Which one?
the prior one.
Huh? This one?
Prior one, yeah. So where did you get this slide from?
It was one of the review articles that I read. I will cite it later if you like it. I thought it was interesting how like each cancers had, according to them, different screening goals.
Oh, colorectal, minimize false negative.
Yeah, I thought it was interesting, so I just put it up there.
Breast, minimize false positive. Ovarian, minimize false positive.
So they were talking about how each cancer should have a different goal, like which is why they were advocating for like singles cancer screenings.
Oh, advocating for single cancer screening because goals are all different.
Apparently, yeah.
Breast cancer, fine needle aspirate biopsy. Target test requirement. So I can understand breast and ovarian minimize false positivity, which is the same thing as, you know, if it's positive by the test, it has to be positive. That's high PPV, but colorectal, try to understand intuitively. Why is it important to minimize false negative? I guess, I don't know. Colonoscopy would take out all the polyps, but you could take out the polyps and miss actual tumor. So you have a lot of false negative, just benign polyp, and you would be assured that, well, I took the polyp that wasn't cancer, and that was kind of if there was a true cancer somewhere else, then you're missing it. I don't know how that would be reflected in blood assay. Yeah, I understand this is a good perspective, but I don't quite get it. Maybe we should read the article and try to
Yeah,
really
I like
understand
I like
what they're
to
trying to
skim
say.
through it. So like I wish I had the details, but I honestly like skimmed through it for like 5 minutes, so I don't have the details. I just thought it would be an interesting introduction to the single cancer screening, which is why I put it up there.
Mm.
But like I said, my presentation is not complete yet. I'm sorry about that.
It's okay.
Yeah. So I'll move on to single cancer detection. One of the things out there was Delphi, um, which I thought was interesting. This is their STEAM. They basically look at machine learning models for detecting tumor-derived cfDNA. Um, they analyze the cfDNA fragmentations. They have a prospective study of 365 individuals at risk for lung cancer. Um, and then, uh, so they basically detected, uh, so basic, what they did was they not only did the fragmentation features, but like they also combined it all. So they looked at fragmentation features, clinical risk factors, CEA levels, and followed it up with CT. And then they said they detected 94% of patients with cancer across stages and subtypes. And I thought this was kind of surprising because like, well, not surprising, but interesting because even stage 1 was pretty high. So
So sensitive. What is sensitivity? Is that sensitivity detection rate 94? So I think what we need to, so when we compare across different platforms, I think we need to make sure we're comparing apples to apples. Yeah, when they're saying just detected 94% at 80% specificity. So I think we need to understand what that really means, how they calculated that. Yeah, because that number seems awfully high.
yeah, this one is pretty high too for stage 1.
Okay. But it's Nature Communication came out last year, so.
I'll
Okay,
double check on the numbers.
yeah. Well, before that, but can you go back? Yeah. So I just want to tell you the science behind, it's called fragmentomics, DNA fragmentomics. What it really means is this company, is it Delphi is the company name?
Yeah.
Okay, Delphi and Lexan Bio or Biodesics or Freenome. These are the companies we're studying, or Guardant as well. They're studying this and they've incorporated a lot of this biology into their platform. What it is DNA is usually coiled in chromatin, and when it's all coiled, there's no replication or transcription happening. So that means that part of the genome is not active. So by studying, and so it's not exposed to any fragmentation. So there's enzymes that will make DNA into fragments. When they are coiled, it's almost like studying RNA. When RNA is in a transcription mode, then you know that's how we study our RNA expression level. That's the study of transcriptomics. But even before that, DNA has to be out as a template, not coiled. So if they're out as a template, they are fragmented. So understanding which part of the DNA exists as fragments and which part doesn't gives us an idea which part of the genome is being translated or transcribed. So just looking at that, because DNA is much more stable than our RNA. So that's how we know biology, what is happening. And just looking at fragments of a cell, we know, oh, this cell is coming from liver. This cell is coming from lung. You can decipher its origin, tissue origin. You can decipher whether this is coming from T cell, B cell. You can decipher whether it's coming with whether the B cell is active or not active by looking at which fragments exist. But that's a lot of information. So definitely you need AI help here. And this is a fascinating biology. And it's just not, and same thing with the methylation pattern. Like it's the pattern and it's not, you know, what gene is methylated, what gene is not methylated. Each individual methylation pattern may not make much difference, but it's the whole global cell methylation pattern. And then it's different layers. It's, you know, protein glycosylation, phosphorylation, protein expression, RNA expression, RNA exome splicing, what RNA is there, and then what DNA is there as a fragment, and what DNA is mutated or not mutated, and what is the copy number of that mutation compared to the germline mutation. So what's amplified, what's deleted. So that whole layer, it's all linked, but it's all different signatures. So if you combine all these layers of multi-omics, then you might need a drop of blood. If you just use, let's say, DNA sequencing, then you might need 10 cc or 20 cc of blood to figure that out. But DNA fragmentomics is a recent breakthrough, and I've been following this. I've published this work in with Lexan Bio. It's clinical cancer research. You guys should all read it. Andrew Davis is the first author. I am the corresponding author. We used whole genome sequencing, DNA copy number, as well as DNA fragmentomics to monitor our lung cancer patient treatment response. So I want you to understand the science, not just say, well, this is better, this is not better, I think the science is really fascinating. And it's really that those scientific breakthroughs that led us to have these technology in our clinic. So yeah, just a few words about fragmentomics. You can go on.
Yeah. So yeah. Then there's Freenome, like you mentioned before. Freenome uses artificial intelligence, machine learning to identify patterns of cell-free biomarkers in blood. They have a pre-MCRC study, which is a prospective multi-center observational study to detect colorectal cancer. They have like around 25,000 participants. So they
Did they oh, okay. Is this
Huh?
their publication?
Yeah, it's their publication.
Okay.
So they looked at 546 colorectal cancer with 271 non-cancer controls. they said that their cohort was heavily weighted towards early cancer, like 80%, stage 1 and 2. And their mean AUC is 0.92 with sensitivity of 85% at 85% specificity. And they said sensitivity increased with tumor stage and tumor fraction, which makes sense. Yeah, and then there's like a lot of studies out there.
Can you go back? Let's. So this was.
Let me know.
Well, I guess there's majority of their patients were stage 1 and 2. But I guess sensitivity of 85% is really good. So I maybe I need to update myself on sensitivity and specificity. Mean, so they report like mean of sensitivity at, remember that, the other paper had at 80% specificity.
Because you have to see them together, is what I thought.
Right, but I don't understand like you can change your sensitivity by changing your specificity. Like, is it like a calibration?
They're like interconnected.
So I guess my question is, this is just it's about epidemiology statistics. Like, can you set your specificity at 90% and then your sensitivity will change? Because I thought you have the true number and the test numbers, and then the sensitivity and specificity will come out from that, 2x2 table. But the
if
way
we
they
change the
Mhm.
if we change the threshold, then the sensitivity and specificity will change according to that. So if we want to increase the sensitivity to 100, the specificity will go down. So it's all depend upon the threshold. So if we keep one threshold higher, for example, we want to keep the sensitivity higher, then the specificity will go down of the test.
So I guess, but at a threshold where you put the specificity to 85%. Okay, and now I understand. So it's not, well, I guess you're changing the threshold to get to 85%, then you change your sensitivity.
Yes.
Like, Right. So yeah, I guess there's a calibration happening as with any other tests. How would you define detected or not detected? How do you cancel out the noise? By canceling out the noise, you might cancel out the true signal. So there's going to be a lot of that happening. So I guess in that way you could change your threshold. Yeah. But still awfully high sensitivity for stage 1 and 2. Okay, you can move on.
Yeah, and then a question that we can think about is that, do these tests have a future? And I guess what company has the best change? But right now, I mean, Gallery has the most trials out there, so the other companies are catching up. So I feel like this is not a question to be asked at this moment. I feel like this should be asked like at least like 5 to 10 years from now, because everyone is still in the developing stage. They're catching up pretty quick, but yeah. Do I think the platform is solid? Honestly, I don't think so. Well, at least not yet. Um, I do think we need more trial results. I thought gallery would be pretty solid, but looking at the articles, I'm not 100% confident yet.
So what made you think that?
Um,
What can you go
I
back
was
to the gallery trial results?
well, one of
Can
the
you?
result was that
Mhm.
like one of the reasons was that they have 4 trials going on and like um I was actually more interested in the um I guess pan cancer would be really nice, but the other article resonated with me where they said one will not fit all. Like um every cancer should be directed a little differently. Like for me it was more about like, you know, the mortality or like the the aggressiveness of cancer. Like I felt that something that would be mortality if the mortality or like the aggressiveness or like the progression part is like higher. I feel like those cancers should be um m- more targeted in a sense. Um and in that aspect, like gallery test, it's great that they can detect a whole variety, but I would like to see their results for the ones they're having right now for breast cancer and lung cancer. I would like to see those results first before I apply anything to clinical practice. Um,
So can you pull up their publication and can you let me know which part you weren't satisfied with?
I guess not satisfied. I just want to see the results, you know? I just want to see what the results are gonna be. The Strive and the Summit trials, because it's still
Mm-hmm.
going,
Mm-hmm.
it's still undergoing. So like they have
Mm-hmm.
published that they are going to do these trials, but they haven't
Mm-hmm.
published data yet. So like
Mm,
I would like to see what they come up with first. Like
mm,
I would like to see that the Galleri test works for individual cancers first before applying it for in the pan cancer setting. And also like another thing for me was the price of it. I guess the
mm,
other screening tests are not available like commercially like the gallery test, but like the gallery test which is available, that's like $1,000. And I don't know if I'll recommend that to like the patients just because it has a possibility of detecting cancer.
mm,
Um and like the detection rate is only 67%. So like well it's still really high, it's still a good number, but like then you have to like balance it up with like, wouldn't it be better to just do the existing tests like colonoscopy for colorectal and like, you know, like the mammograms for breast? Like, wouldn't the existing test better detect? Was my concern
mm,
for the price. Like, I don't think it's cost effective for the patients.
mm,
So I think it has a future, but I wouldn't apply it to the clinic at this point.
mm.
Unless it somehow goes through insurance and somehow the you know insurance companies decide to pay for it, then I guess it's not a problem. But like compared to the price, the benefit to the patient is, I don't know, questionable.
Mm. Okay, price set aside, let's just look at the scientific validity. This is not a, so only data we have is CCGA, and this is based on, pretty large cohort, 15,000, and they were kind of divided half-half to newly diagnosed cancer versus control.
Mhm.
and more than 50 different types were there. Thinking there were sub-study. And is this part of that, or is this a different validation cohort, sub-study? Probably then, or is it? I'm not
The
sure what
The
it
results
means.
are from the sub-study though. I think they gathered all the people there, but the results down the preliminary results are from the sub-study as far as
I think they got sub-study population with some eligibility, I guess, or what's analyzable at that point or something. Okay. Anyway, they selected 12 types, and so they looked at just 12 types, right?
yes.
They didn't look at outside of this.
No.
Okay. And specific, so if the signal was there, then 99% was true. Tissue of origin was correctly identified. So I think this is remarkable that fragmentomics can detect tissue of origin, 93% accuracy. Like, you know, with just the blood, let's say 10 cc, they 20 cc or something, they can say you have pancreatic cancer in your body. I think that's remarkable technology-wise. We didn't have that technology before. And it's correct 99.3% of time. Again, this is not a prospective study. This is case control, but prospective collected sample. The test had 67% detection rate. And this is just for 12 pre-specified cancer alone. So this will only go up as you add more cancer type. And this is stage 1 to 3. And I don't think they have, I think they included only 2, 3. They don't have stage 4 cancer here, or?
No, not for this study.
This cohort. So I guess if you include stage 4, the rate will go much higher,
Yes.
you know, as you expect. So just for across these tumor types, and pancreatic cancer stage 1 can be very, very small. So detection rate is 43% across stage 1, 2, 3. And I think, well, I guess this type of case control, and this is, you know, and this is just that if this is just a subset, this is 6,000, let's say 6,600 side study, and not a prospective data, but case control. And as a society or people majoring in health economics, it's their turn. Policymakers, finance people, it's their turn to make informed decision. Am I going to reimburse this test or not? Now, by providing this test free to my employees, will I be exempt from paying their cancer treatment costs by early detection? You know, I won't have to pay future chemo radiation costs. We know how expensive those tests are. Even Guardant, Tempus, all these tests I order, they're like one blood draw is like $6,000. Would this 990, and that's before reimbursement. I think when they reimburse, it will be a lot cheaper. Will this early detection, cost of treating stage 4 pancreatic cancer versus stage 1 pancreatic cancer is way different. So understand that no test is perfect, but if I can detect asymptomatic stage 1, 2, 3 cancer patient which can lead to cure and not indefinite like chemoradiation, is this test worth it?
I think
Now I think the prospective study has to answer this. Why the reason I was very surprised that Galleri test is being sold in clinic is that I never knew that 6,000 patient case control study data would lead to commercialization of such test. And there's a lot. So it's not, it may not make it to the mass public, but there's definitely a high demand. People where money is not our issue, they want everything to be done to make sure there's no cancer in their body. They need to live long. Can you invest $1,000? This is out of pocket pay, but again, I think the payers will quickly reimburse these tests. And then the second question is mammogram, colonoscopy, low dose CT for smokers. These are proven ways to detect to do an early detection. Let's say adding blood to these tests adds no value. I think that's fine. Even if it adds 0 value, I think if it can work to detect ovarian cancer, pancreatic cancer, colorectal cancer, gastric cancer, esophageal cancer, biliary tract cancer, the cancers that there is no way to screen them with current technology, I think it still holds a lot of value. And head and neck cancers, thyroid cancers, prostate cancers, which there's some of them do not have PSA or other tumor markers associated with it. So there are still ones with very high unmet need for screening.
Mhm.
If this test can show its utility there, I think even if it fails in majority of like the common tumors where there is already a screening tool, I think this test still holds value. So that's
I guess.
my clinical thought.
Mhm.
Mm,
I think
but
for
again,
me, it's not.
mhm.
I think it's just partly because I haven't gotten out of my Korean medical perspective
Mm,
is my one of the reasons why I feel this way, because I guess I have to start understanding the American medical perspective, because in Korea medical services are very, very accessible. If you cut your finger, you go to the hospital. Like it's no big deal. Everyone gets screened. It's universal healthcare. Um, so I guess it was like it just didn't make as much sense in that setting, because I feel like in Korea there's uh everything is already very accessible, so they would have already done all the existing tests anyways. So another added test will uh not affect it as much, was my thoughts. Um but yeah, I do understand what you're saying though. I don't know, I guess I still have very conflicting thoughts about this.
so I think when we look at tests like this, we have to know where we're coming, what's our background. we had 0 way of screening these tests, these tumor types. CA-125 has never proven to be a good way. So this is, I think, the first time that we are now equipped with technology to detect, lethal cancers at early stage with just a tube of blood. And there's a difference, blood draw and actually having to prep to go through colonoscopy, coming for mammogram, low dose CT. These are different. And then I think blood will only help. Even if you find nodule, then what are you going to do? I mean, there are next questions, and should we do a biopsy of that nodule or not? So, and blood tests can help aid in that decision-making process. So I think in your review paper, I think we have to emphasize that not only aiding in the screening options that we have, like low-dose CT, but also understanding the unmet need and how that is, first realized with this liquid biopsy. I think that's number 1 point. Number 2 is the tissue of origin. I think this is fabulous. Just the blood will tell you what tumor is in your body, and that's pretty accurate. I think it will. I think it will help in our therapeutics. Let's say lung cancer, you do biopsy 3 times, you get hemothorax, pneumothorax. I can't get any tissue, comes back necrotic. I don't know what type of lung cancer this patient has. I was so frustrated many times, and I have to treat blindly. I only know it's lung cancer, but these type of technology will tell me this is neuroendocrine tumor. This is kidney cancer spread metastasized to lung. This is small cell lung cancer. This is non-small adenocarcinoma. Tissue of origin and tissue subtype can be easily identified with this technology, which is amazing. I think this is breathtaking. This is a new technology, and it can tell you. And what's amazing about Freenome's test is you can look at the DNA just like a single cell technology and say, oh, this is a T cell, this is B cell, this is liver cancer cells, because you're looking at the whole host cell-free DNA along with tumor DNA. So that sort of lens, understanding the immune landscape along with the tumor landscape, everything else all combined. It's almost like tissue specificity and where everything lies. So that technology is amazing and has infinite applications. And I'm just amazed at that aspect. And then I think the detection rate has to be compared. Well, I think Freenome had really good test, the compared to the colonoscopy. I think the Freenome's test had added value there. So okay. So those 2 points should be discussed, and like you said, prospective data is lacking. Maybe once, but then those are, one is breast, one is lung.
Mm-hmm.
And I think they have a, Pathfinder, which is the multi-cancer
Mhm.
perspective. So I think at best they're, because it's a cross-sectional, it's like they're probably, what's the annual, because their endpoint is within 1 year, what's the chance that what's detected in the blood will be found within that 1 year. But it's not like a lifetime cancer risk. It's still annual, so it's in that way it's cross-sectional. So I think the highest in general population, maybe age of 40 to 50 above, would be maybe 1 in 100, maybe in totally asymptomatic patient, not even patient, but population. So yeah, I think it's likely to be positive but it's almost like what is our threshold as a society? Like how, at what detection rate are we ready to invest?
Mhm.
And that's really a discussion of policymakers and health economists. But in a society where medical cost is really high, America spends a lot of healthcare dollars. So I think America is more motivated than ever to invest in technology like this, because this is a country where you do anything to keep people out of hospital. They do care round. So Junie, tell us about the social workers and the care managers. What are they trying to do? Tell us. Junie? Is Junie listening? Maybe she's busy. So they're trying to kick people out of hospital as much as they can. So they harass residents every morning. Like, why can't you discharge this patient? What is the rationale? Okay. Every morning. Okay. It's your duty to report to case managers, because hospital loses money by keeping them longer than they should.
Mhm.
So it's a very cost sensitive structure. And if they can reduce amount of cancer patients in any way, requiring advanced treatment, they'll do it, because those are so costly. Cost of chemo, CAR T cell therapy is like $1 million per shot. So, so if you can, it's the cost of therapy versus cost of detection is no comparison. So they'll do all these calculations to figure out what's more effective, cost effective. But I'm sure early detection is what they will invest, and I can see these tests become. It was like that before. No one was using Guardant or Tempus, and they say it's not going to be reimbursed. It's like NGS for all solid tumors. Doesn't make any sense. It has to be breast tumor panel, lung cancer panel. Look, Guardant and Foundation Medicine, Tempus, they all got FDA approval for all solid tumor sequencing. People thought it's not possible. So I think the next wave is the general population. 23andMe is commercial. A lot of people use it. 23andMe. Now this is for cancer detection. This is not just exploring your genealogy. And people will do whatever it takes to know their risk. And they know they have loved ones died of pancreatic cancer because it was discovered too late. And if they find out that there is a blood test with decent validity, So I think that's why we need a good paper attesting to its validity and where it stands in human history of cancer detection. So I think we have to talk about scientific validity. Where are we? And then as a society, are we able to accept it or not? It's really the job of health economists or policymakers. But at least the scientific and the medical historical significance and where we should be headed, and where do we foresee the future coming in next 5 years is sort of our job. But I'm just still amazed at how fast this technology has come forth, into clinic. So, all right, Jui, good job. I would like to see more in depth, like where the trial data, and maybe compare. So this is not going to be just for the presentation. This is, how do you compare each technology? I want to see some comparison between different platforms, number of samples, perspective, is a case controls perspective. And you don't have to do, I think people will want to know the future. So I think we need a two table for your review paper and for your next presentation in the lab. So one table would be for what's completed and published or presented, comparison between each major trials or studies, whether it's case control or perspective. And then the game changer or pivotal trials that are ongoing. What are the commonalities or differences between trials, technology, number of patients, population, platform, and primary outcome, and the comparison and all that. If we can make that into a table for those completed and ongoing trials, that would help a lot. And you can work on it as a team.
Yeah, I'll keep that for the future.
Okay. Updates on where you are on your project.
Hi, Dr. Chile, can I share my screen?
Yeah.
I'll just do it after Arnie and Sumi does it then.
Sorry, Juhi, did I interrupted you?
No, you can go on now. It's okay. Go on first.
No. So this is a table.
Did we put discharge on this one? You did?
No. So this is the final table that we have made. It has candidate 1, tumor size 1. That was the total tumor size of all the target lesions at that date. And then scan date 2 and tumor size 2, sum of all the longest diameters of the target lesions. And then scan date 3 and then tumor size 3. And then we have responses. So Dr. Minjong will use R script after this table to calculate the hyperprogression.
Okay, the table looks fine to me. Maybe work with Eugene to put volume here as well.
Yes, the only problem for the volumetric data is that they don't have any patient identification information. So maybe I think I have to ask Yuri to provide that to match with the existing data.
So was there a patient number? Patient 1, 2, 3, 4?
Yeah, there is only artificial number, just 1 through 200. Other than that, no MRN, no clue to guess who is he.
Okay, just CC me and email Yuri. There may be, yeah, there.
Got it.
Yeah. And also at each 3 different time points, we need volume. So
Okay,
make sure you have all that volume at each time points. Okay, I think this is good. So does a blank mean that the scan wasn't done, or what do you?
Yes, so for
Oh.
0, it's like it's not done. So whenever there is no date and there is 0, that means that the scan was missing. So we are planning to make the flowchart and then we will remove the values with 0. So we'll exclude them.
Okay, so
Actually, in the down of the table, I already count the exclusion of the number. Go down. Like
I'll go down. So there are 66 patients with no follow-up CT, no baseline CT, and CT outside images. Immunotherapy patients which did not receive immunotherapy are 16. Patients with no target lesion are 9. Patients with post-lobectomy are 11. And other than that, there are 4 other. So we'll try to make an flowchart for it. And then we'll add it in our paper and abstract.
okay, and for immunotherapy, I think you should differentiate monotherapy, combination immunotherapy. They're still in the category immunotherapy only, whether it's single or dual. And then there can be chemo plus immunotherapy. So I think we have to differentiate what they received. And that data is collected by Dr. Yoonho Lee. And it was part of his prior abstract coming from the same database. And his conclusion is hyperprogression rate is almost 0 if you combine chemo to immunotherapy. So I think when you analyze the data, you do have to analyze different subgroups according to therapy.
So do we have to add one more column to this table to check that if they were on immunotherapy only or if they were on immunotherapy plus chemotherapy?
So I think all these clinical treatment variables were already collected by Dr. Yoonho Lee. So you have to merge Dr. Yoonho Lee's data with what you have.
Okay, sure.
And Eugene Oh and Minjung Kim can help with that process.
Okay, thank you.
You're welcome. Good. Yeah, I think this is great. Still a lot of tuning up data. All right. Anything else? Junie and Arnie, anything else to add from your mutual project?
No, not from my side.
Okay.
No, for me it's also. Doc, I have one question. So there is for the exclusion criteria. I already counted, but there is some patient that we only have the pre-baseline and the baseline. We don't have the after treatment one. So I exclude it. So should this data I still put it on the table or should I just delete it? because we didn't count the RESIS criteria because we only have the pre-baseline and the baseline one.
So this is not like we're waiting for the scan to be done. This was not done for something. It's like treatment started, patient passed away. Is that kind of situation, or was it too recent?
Usually if the CT not available, if not a CT outside image, or if the CT like more than 3 months.
Oh, more than 3 months. Okay. Okay. Yeah. No, that should be in the flow sheet then.
Okay. So I won't delete the data in the table then. Okay. Thank you, Doc.
No problem. And the other thing is, we're looking at with the lens of hyperprogression here, but all the data you have will also be utilized for what is the best radiomics feature to determine predictive power in patients receiving immunotherapy. So not the pre-baseline treatment baseline scan. Even if that's missing, if you have baseline and post-treatment, I think we would still wanna, keep that data. I mean, we know who they are, but I think that those are the only 2 scans we'll use to answer that radiomics question, apart from this hyperprogression project. So I just want you, Minjung and Eugene specifically, to keep that data where even if we're missing pre-treatment, as long as we have baseline and on treatment, keep that data. And Eugene, we should send that data to Dr. Yuri so he can use it to analyze radiomics features. And that's the data we'll be sending out to Lunit as well. Okay.
Okay, so you mean that if we collect all the clinical and then hyperprogression related information and the volumetric data into one file, we should do that and send it to other parties as well, right?
Right, even the ones without the pre-treatment.
Okay, even the, okay, gotcha.
Collect that. And that's the file we're going to be sharing with Lunit and Dr. Yuri. Okay. Yeah, I mean, we will still include pre-treatment information that we have, but it's not like we're going to exclude those patients from our list. We will still have them in our full list for radiomics project. Just for hyperprogression, we need all 3.
I have one question, Doctor. So we will make 2 copies of this sheet, right? For hyperprogression, we will exclude the patients without pre-baseline CT. But for the other project, we'll make a copy of this sheet, and that copy can be used for the other project, right?
Yes, I think that's ideal. Yes. Okay. Any other question? Okay. We'll go back to Juhi.
Okay, 1 second. Sorry. So for the review paper, everyone was a little busy this past week because everyone was wrapping things up. So we are not done with the secondary screening yet. We're done with the so secondary screening has two parts. It has the selected articles part and the undecided articles part. All the groups are done with the selected articles part. And they will finish the undecided articles part today. So by the end of today, before everyone goes home, I hope to receive everything, all the secondary screening done. So after today, I will go through, I will compile everything up, and then I will assign full text article, and then we will talk about the final articles that we did choose, and I'll assign the articles to each person and then ask them to extract information from the article into a table. And for that extraction of information, the undergrads are gonna help out too. So there's that. Upon screening, so one of the things that I realized was that there are a lot of articles out there. And if you remember, like for the presentation, how I divided it up to multiple cancer screening and single cancer screening, I feel like the review should do the same. There are too many articles to do both. So I feel like for the purpose of this review, it's better if we just focus on the screening tests that do it for multiple cancers, and then maybe have a follow-up review with single cancers, because there's just too many articles. And yeah, so I was wondering what you thought about that.
Yeah, I think it's going to be a lot of data.
Yeah. So even for multiple cancer, the screening, there's at least 15 from the selected part, but we didn't even go through the undecided portion yet. So I feel like this review paper should just focus on the multiple screening for now, and then have a follow-up review. I could work with someone who comes after me. I could keep working with them to go for like the single cancer review, if that's okay with you.
That's fine. I think makes sense to do multi-cancer, and then within single cancer, we can make it single cancer, but we can make it, selected cancer type, whether combined or if it's too much for one colorectal, we could do colorectal review. I just don't want it to be, a lot of work for you. So, I mean, you're welcome to continue as much as you want. But I guess the priority is multi-cancer for sure, because I do want, like I said, lung cancer, breast cancer, we have technology to do early screening, but I think focusing on what was previously missing should be the priority. Number two, I don't know if you covered all the companies. So I think we should cover not only the companies that has the completed study result or any preliminary result, but I think the companies were claiming that they have the technology and they have some sort of, I mean, if they made the company and they got investment, they should have some preliminary data. No fool will invest their money on a company who has, zero data to show to their investors. So you have to believe there's some level of data So including company like Predition, whom we met at the AACR meeting, any company that is in the market, I think we should mention them or have a table of who are the
but Precedent was MRD.
the companies companies or laboratories in the in the market, and what are the preliminary data that they have, and what kind of platform they use, and what are the prospective trials that they are going to report soon. So I think we need a comprehensive review about those players.
Well,
and that's more pertaining to the multi-cancer screening. In single cancer, it's still important, and I think I think the future would be that we will have one blood test, but it will use different algorithm. It's still blood-based, but the algorithm used to predict risk for lung cancer would be very different from algorithm used to detect stomach cancer. So it will be individualized that way, but actual risk assessment, I think we'll have a clinical risk model, just like CHAD score or Corona score. You know, lung nodule detect from X-ray, CT, plus the blood says something, and you know, I'll plug in all the clinical variables and come up with the composite risk score. So those things will happen as these blood tests mature in the market. So yeah, I think it's a very interesting discussion where individual cancer risk will be very different, even with the application of these liquid biopsies. Okay, so for now, that we could focus on multi, and then but you could still collect which are kind of finalize what are the single cancer detection articles, and then we can kind of save that for the next paper.
Yes, and for the paper, our selection criteria was a minimum of 30 patients and 30 control. Because I feel like if it's less than that, then it's too little.
Right.
Yeah, but that was it for now.
So what was your like as you searched articles, what are the case and control numbers that were more like common?
It was at least like all the studies that felt good was at least like 50 and above, like cancer patients and control. Most of them were in the hundreds.
Okay.
So this is us going through the first pile of note articles and deleting or putting them in undecided. So this is everyone's input.
Okay.
Yeah, and then I just wanted to talk to you about information to extract from the articles for a table.
Okay.
Like, because this was some of the things that I thought would be nice to have in our table, but
we should discuss, definitely important thing to extract. So let's get back to it. Let's have a bathroom break, and we'll return in 5 to 10 minutes. Okay.
Okay, let's go.
Okay.
How do I make this big again? How do I make this big?
patient issues and schedule issues. So yeah.
Thank you for your hard work.
Yeah. Is Juhi ready?
Yeah, so this was my question. Like, what you thought about um what other information would you advise to be extracted?
Yeah, so the perspective, is it a population-based study or is it a case control study? And what type of cancer are they aiming for?
Yeah.
Sensitivity? Maybe if it's a population study, positive predictive value, negative predictive value. Um, RNA, protein, or did they use fragmentomics? Did they use methylation patterns? Did they use, so a lot of different platforms, and did they use combination of those? So you can put yes or no, and if it's combination, what is a combination of? So just, write all the different technologies that you can extract from each article. There, glycoproteomics is one that InterVen is using. Is it just proteomic signature? Is it combination? So CancerSeek is an example where they do a combination strategy. So what is it? What is the platform utilizing? So there's a laboratory called CLIA Lab, CLIA, CLIA. So CLIA certification is important because this is a blood assay.
CLIA?
CLIA, CLIA. Or if they claim to have, sometimes they claim there's a New York State laboratory certification or something like that. So all these blood tests will claim to have, they are certified by something. So that certification status is important. I thought that, there's no single test that's FDA approved yet. So I don't think FDA approval is important, but I guess whether it's commercial or not makes sense. That means for it to be commercial, it has to be certified. Like the quality wise, it has to be certified, and they're under mass production that when you pay it, you can buy it. Whether it's research use only, like I want to use it, in my laboratory, whatever research, whether it's research use only or not. If it's something you can order and buy, that means it's commercial. So whether it is commercialized or not is another category. Anything else you can think of? I think we have to be very thorough because you don't want to go back. I guess if it's a prospective study, then you want to write down all the characteristics. Like what's the follow-up period, where, what location, was it multi-location or is it one country? And or is it all stage cancers, or was it stage 1, 2, 3? Yeah, I mean, important traits about each study. Yeah, I guess multi-cancer, and we're not, I mean, in your presentation and in this review, we didn't even go to the high risk group yet.
No.
And that's its own category. But I think we'll focus on general population first. High risk, because some studies will look at, but age, we're gonna say age is high risk, but I think for the purpose of this, if you limit to patient age of 60 above, I think that's fine, as long as so you describe what the characteristic of the population or case control population that they looked at. Yeah. Anything else from anyone that we're missing here? The categories. Yeah, I think this is pretty much it.
Okay, sounds good. So yeah, we'll finish up the secondary screening by today. I'll compile everything, and then I'll tell you what the final numbers are tomorrow. And then I'll assign the articles and we'll start extracting information out. I will finish reading the review articles that I've collected. And yeah, so like this week is mostly going to be extraction of information.
Okay. So I think with the review articles, you can kind of point out what are the points that needs to be discussed. And you can present those discussion points next that meeting, what they are. And we can talk more about it.
Okay, sounds good.
Okay.
Yeah, that's it for the review article. For the case report, Vivian Panu, Artie is working on finishing up the diagrams. So the CT scans, the timeline. We're gonna have a timeline too. Like Albert Curie. So yeah, Artie's working on that. We're trying to go a little fancy with the timeline, so it's gonna take a little bit. It's gonna take up today. But yeah, we're doing that. For the introduction and discussion part, Eugene is doing an amazing job reading through the articles. I will try to help her out in that too.
Is
Yeah,
that
because we have to reread everything now. So I was a little.
is that what you guys drew?
Is that what? No, this is just for reference. Just an
Okay.
attempt to understand how the genes, how everything's are working. But yeah,
Okay.
no, but do you think it's necessary to include something like this, or would everyone already know? But since we don't need to. No, this was just for our understanding. But if you think it's necessary for us to have a diagram that looks like this, then we will include one.
Mm, I don't think it's necessary. I think we should just describe that
Mhm.
how P10 feeds and loss of function feeds into the mTOR pathway activation. I think that's all.
Okay, sounds good. Our figures are just CT, PET CT, um, and timeline of events and garden Signatera.
So try to combine as much as possible. PET and CT can be one figure combined,
Mm,
whether you want to put it in A, B, C, D, or, you know,
yeah.
and then MTM time of events.
I was gonna combine the timeline with Signatera,
Yeah,
but
that
we'll
maybe,
see how
yeah.
that goes. Arnie is going to try to see if that works out. What
Mhm,
she's looking at is like super fancy, but we'll
mhm,
see if it looks okay.
okay.
Yeah. Figures should be done by today. Uh, research into the current literature is gonna take this week
Mm,
because we have to redo everything. So
mm,
we're taking a step back. Yeah.
mm. Okay, very good.
Nothing else for me.
Okay. Let's move on to Eugene.
Okay. For me, the one thing that I wanna give feedback on is the our case report for Albert's query. So
Mhm.
we finished it. I think we finished it, all the things we have to do. So let me share that first. Um, can you see the screen well, or do you want me to put the link on the chat box?
Oh, just ask me questions.
Okay, this is the whole manuscript that we prepared.
Mhm.
And could you have a fast look at it, or can I just ask a few questions? It's kind of ready for submission.
So maybe, yeah, just give me a link, then I can skim it through with you.
Okay. Yeah, I sent the link in the chat box. Please check it out, everyone. See if it's working. If not, please tell me.
All right, you guys see my screen now?
Yeah, we can see.
Okay. So which journal are we aiming for?
JTO, and they have a word limit as 1,000 words. So it's
J,
kind
J,
of, yeah, JTO.
JTO.
Yeah.
So you can't use IMA. You have to put IMA here. So does JTO require abstract? for
Yes.
case report?
Yes.
Okay.
They
No
require
evidence of disease recurrence and no evidence of organ rejection. Okay. Why do you have the star?
Oh, so as of now, I didn't put the citation. I just marked the citation, and then after finishing it, after having confirmation from you, I'm gonna add the real citation.
Clinical trial
Do you have to
with
uh-huh.
with a EZH2 inhibitor is better.
Gotcha. Okay.
Consideration was given to bilateral transplant and lack of systemic nodal metastasis.
Oh,
This
so
doesn't make any sense.
yeah, true.
Given the lack of the additional medical therapies and the lack of systemic
Okay, I'll move that.
no metastasis. So did anyone reread what you guys wrote?
Well,
Is
I
it correct?
Sorry, I didn't have time with the because of the cancer screening, but I'll do it today.
Okay. I want the writing to be publication quality. Otherwise, I'll be picking on small things and grammatical errors and, you know, like sentences like this that doesn't make any sense. Mm. Okay. I don't know, low response to chemotherapy. This
there
does not.
there is an evidence for there's a like some actually the citation one has the same contents for chemotherapy, but I'm also not sure about immuno and targeted therapies. But for chemotherapy, it says IMA showed not that good response to chemotherapy. There is a sentence like that in the paper.
Mm. Yeah, I haven't seen anything about target. So that's a general. So, you can express it like low response to systemic. It and with, I don't know what the best word is with.
Okay. I'll take a look at the article itself and then modify it. But I'll definitely delete immuno and targeted therapy because I haven't seen it in anywhere. So, but for chemo, there is something we can send.
Yeah, or you can just cite the chemo article and say systemic medical therapies or something. Just make it more general and then cite that article.
Okay.
And then because you're citing what's been reported with known low risk, with known, with its overall poor prognosis due to. So if you say listed, I know you're citing it, but you have to say, what guideline.
Oh, okay.
You can't just say listed. Use of cardiopulmonary bypass. So here you describe it as someone else used it, but it's Northwestern, us writing this paper. So you have to say, we use this technique. We did transplantation for COVID-19 patients. So it's okay to use we, okay?
Gotcha.
So is this the garden you wrote?
Yeah. And the table is in below. In the actually table was made by Juhi. Thank you.
We will synchronize the styles later.
Okay. You say the clone here.
Yep.
So you say MRD was absent using an assay based on additionally. So
Yeah, maybe do we have to mention about garden in the case presentation as well?
yeah, so this is not only pertaining to this paper, this is also writing 101. Any objective finding should be in the results or case summary or whatever that is. Discussion is discussing what's already reported in the results section.
Okay.
So what's found from the garden essay should be all described prior in the prior section. And discussion is discussing that finding.
Makes sense. Got it.
Okay. So I think we have to find our stance here, which is because, so this is what I would emphasize is very important. So I would say, tissue validation is important to remember this. So Signatera is important because it used tissue to validate what's found in the blood, what's truly coming from the tissue, resected lung cancer. So this test showed that there's no trace of cancer in the blood that was found from the tissue. So we can kind of emphasize that even in the abstract, that tissue-based assay did not reveal any MRD. However, Guardant, which is not tissue-based, so I don't know where TP53 mutation is coming from. I don't know if it's coming from his lung cancer. I don't know if it's coming from his MDS clone in his bone marrow or colon polyp, which is colon adenoma. So I have no idea where it's coming from. And I have no idea why it's still present when Signatera assay is still calling it non-detectable MRD. So I would still emphasize tissue-based approach showing non-detectable MRD, but I would gently mention Guardant result, what it means in discussion. So you would discuss, you would report what you found in Guardant in the result, but you would discuss, I don't understand why Guardant is still showing up while Signatera is not showing. However, I cannot make any comparison because Guardant, Signatera is not giving us, I think Seungmi knows, Signatera is not giving us the 16 gene mutation because they think it's proprietary. So I don't know what those 16 genes are, but that those genes are claimed to be a unique mutation from the tissue, and we cannot find that anywhere in Albert Curie's blood. So I think it still is scientifically meaningful finding. But the Guardant, you can argue that there is still lung cancer clone detected, but this was not proven by tissue validation. So I don't know whether these clones are truly coming from lung cancer. The paper, I think it was a Science paper, his first tumor heterogeneity paper, Charles Swanton, you can find it. It's about tumor heterogeneity. And what it showed that the tumor allele frequency in the blood correlates directly with the tumor volume. So for instance, 1 centimeter tumor would correlate with 0.04% allele frequency. If it becomes 10 centimeter in size, which is the volume wise, that would be a lot more. That would be about, let's say, 4% or 5 or even 10%. So it has shown direct correlation with tumor size with the allele frequency. Now, Albert Curie had tumor full of cancer. So these are all good discussion points. Tumor full of cancer, which we could assume that if it's truly coming from lung cancer, then variant allele frequency would be very high. And we would expect after removal of tumor, so he has no evidence of cancer in his body, at least radiographically, then we would think compared to pre-surgery Guardant, a post-surgery Guardant, at least ctDNA mutation variant clones. But I don't think it showed that. I think it was still.
Professor, you got cut off. Could you repeat the last few sentences if you remember?
Sure. So
Sorry.
pre and post surgery, there wasn't much difference. At least the level of ctDNA that was detected. So I would argue that if it was truly coming out from lung cancer, then we would have seen a steep drop. Now, however, I think we have a tissue validation from Tempus. Do we have Tempus tissue result? Was that described, Eugene?
No, I haven't. I only described Signatera and Guardant, so I'll look it up for Tempus.
Oh, that's very important. I think you need to describe everything about tumor.
Okay.
Its unique characteristics, pathology. Was it lipidic growth? Was it bronchoalveolar pattern? What was the PDL1 in tumor cell, immune cell? And Tempus, what was the tumor mutational burden in the blood, in the tissue? I think we want to know everything about this tumor. If Tempus has any RNA sequencing, then I want to know what's overexpressed, what's underexpressed. I want to know the copy number variation and what was discovered in the Tempus tissue DNA sequencing. I want to know if that exact variant, not just p53, but it has to be the exact variant, is that same clone that was found in Guardant. That way we have tissue validation. That way we know for sure these mutations are related to lung cancer, and in which case MRD is really not non-detectable. It's only non-detectable by Signatera platform, but comparison between Tempus and Guardant would show us that MRD is still positive. But the next question you need to discuss is if MRD remains same level at VAF, does that mean that it has reached a immune editing and immune surveillance or immune equilibrium that that doesn't necessarily portend recurrence. Okay, so I think it's important to describe this even with immune suppression. Is there a possibility that immune system can still clear things up? And I think we discussed it, Eugene O, that in our ctDNA,
Yes.
there was a case where it was initially detected, but it was cleared up.
Yeah, so in the very last paragraph of discussion, I cited that abstract, and then I wrote about immune editing, and that was actually the last part of the discussion.
Okay. This one?
Yes.
So 42%, can you? So it says after curative treatment.
Yeah, so
you have to clarify whether that was surgery or chemotherapy, because people would think that maybe chemotherapy cleared it up, not the immune system.
mm,
So what does it mean to clear up? So when you want to say it's your immune system that it cleared up without any therapy, that's what, do you understand?
yeah, I have to find whether those patients received chemotherapy, like post-surgery, after surgery. Yeah, maybe I have to make this sure, make that clear.
Yeah, so, and if sometimes surgery can clear up because the cancer is gone by surgery. So you can't you have to have a blood draw, both
Oh.
post-surgery and both post-chemo, post-surgery and plus minus chemo. So there has to be no intervention between the two blood tests. You have to show that it cleared up by itself.
Okay.
And I think the 42% is with curative treatment that cleared up,
Yes. So 42%, the way I calculated is actually, I only add up the patients whose pre-surgery ctDNA was positive, and then I counted only the patients whose ctDNA was converged to negative. So
that's clearance by surgery. Surgery cleared it up.
oh, okay, yeah.
Surgery will take out the cancer, so there will be no cancer in the body. If it was truly only in the colon, right?
Yes, but I think everybody got surgery, but weirdly, like some patients still had ctDNA, a positive ctDNA, and some patients showed conversion. So I think if you can give me a time, let me pull up the data.
Sure. If you have the slide, you can share your screen.
I will share the slides.
Still raining.
So the abstract that I referred to is this. So they all got operation, but still they were able to classify their patients into 4 groups in terms of ctDNA clearance. And the thing I want to write in the discussion was I only focused on the initial ctDNA positive group, like noted in orange and then dark blue. Among that patient, I don't know exact mechanism or reason, but some just for some patients, ctDNA converts to the negatives. So I thought we can cite this part in terms of maybe there might be some act of our own immune system, because they all got surgery, but there is a still difference. But still, I need to make it clear that those two groups of patients may be different in terms of post-operative adjuvant chemo or something like that. I haven't checked that yet, so yeah, I will.
So you have to know when was the positive.
Oh. Oh, it's here we can know that from here. The time point were 4 weeks after surgery and 12 weeks after surgery.
Do we know if any of them received chemo?
Actually, I have to look it up. That.
Can you go and see what did they describe the population?
Oh yeah, I can find the abstract itself.
So it's not published.
Yeah, it's only abstract. So maybe we may not know it if they don't describe it. Sorry.
So you can't find the abstracts in PubMed unless it's published in somewhere. You have to go to the GI ASCO abstract site.
Here, I think I find it. So among total 1,365 patients, ctDNA clearance rate at 12 week was significantly high in adjuvant chemo.
Versus non-adjuvant chemo in stage 1 to 4. So there, the 7 patient out of non-adjuvant chemo cleared up.
Yeah.
I wonder if that group, still 7 patient had good prognosis, good outcome. 24-week is higher. So that 7 patients still remained cleared even at 24 weeks because it's still 7 there.
Oh. Out here.
No. Oh no, it's 0%. Wait.
Yeah, I think it's 0.
So I wonder what that means. 0. So I wonder. I wonder what happened to that 7. Is that 7 all gone? Or not, or censored? Because I
Can I
don't know if
ask?
it was.
Yeah. Can I ask like directly to the authors? I'm not sure, but
Yeah,
feel-
I mean, that's why you have corresponding author
oh,
to
okay.
ask questions.
Yeah.
So yeah, 0%. I don't like that, but stage 2 to 3, still 0%. So cumulative instance of ctDNA clearance was, they put it 7%. So where did that 7% come from? Wow.
Yeah, by 24. Maybe they accumulate all the data. Like I'm not sure. Maybe I can ask them.
Maybe they got 7% from that for at 12-week was significantly higher in stage 1 to 4. So that was 8%. Maybe they just combined that all.
Yeah.
The seven patient out of so 12-week clearance. And non-chemo MRD rate.
It's interesting. Even for limited periods, maybe our immune system can handle minimal residual disease without chemo, but they ended up being defeated by cancer.
So you can, the question can be, all these, so it's 12 weeks. So they were cleared 12 weeks, but were they all censored? What were they all censored, or were they all turned into cancer? So is that why it's 0 at 24 weeks? So that may be your question.
Yeah, I will ask to the author.
Yeah. Anyway, I think you can still cite that 7 patients out of 93 had ctDNA cleared at 12 weeks.
True.
Even if they were positive at 4 weeks. You can, yeah.
Okay, without any adjuvant chemotherapy.
Yeah.
But the thing is, Dr. J, like JTO only allows us to write 1,000 words and with only 5 citations. But I think this case has so many things to discuss. So I already exceeded the number of their citation limit number. I already had 7 without Charles Swanton's article. So, maybe do we have to change our target journal or
I think you would have to be very,
Concise.
so I think you just write it out, what you want to discuss, so that
Uh-huh.
you can use it for other journal. But
Okay.
then really chop out, like, you don't have to write easy, so anything that does not contribute to the main idea. The main
Uh-huh.
idea is, transplant can save.
Okay.
Then you don't need to talk about the patient received, prior lines of therapy, including so. That's it. You don't have to say that. So you can still bypass a lot of it and say patient was under, immune suppression, with tacrolimus, MMF,
Oh, you mean that I can reduce the amount very much
of this,
in case
yeah,
present?
specifics, yeah, about how bad, mucinous is. You can make, I think you can shorten things,
Okay.
but I suggest you write it first, all the ideas you want to discuss, then get to the 1,000 limit. I know it's hard, but I think it's better to do it that way.
Okay. Just prepare another version for JTO and then leave the
Yeah,
remain.
and
Okay.
then you can decide what are the main points, what are the 5 most important citations.
Okay.
And then you can, like, if you have to limit to 5, then you can cut out the ones that are saying the mucinous is bad, because you can take out that citation. I think JTO is a journal both thoracic surgeon and medical oncologist read. So I do believe
There
that
will
should
be
be our
included.
first choice.
Okay.
But the longer version, I think you could target to. So did you ask other journals?
Right. So PLOS Medicine, they showed favorable response. They returned to us like, if you prepare your manuscript, please submit as soon as possible and let them know, so they can bypass any unnecessary process. And they don't have any word limitation, so maybe after submitting to JTO, and if we got results as a rejection, then we can shoot it for we can shoot for PLOS Medicine.
Okay. So I think to reduce the amount of work, submit to PLOS Medicine first. It's okay.
Okay.
They have really good citation score or something. Yeah, try PLOS Medicine. If they were open to it, then submit to them first, and then if they don't take it, then JTO.
Okay. Yeah. Thank you. So in that case, I'll prepare the whole manuscript, according to the PLOS Medicine guideline, and then prepare to submit there.
Let me finish looking at
Conclusion?
tape. Yeah. Let me share.
Yeah. Can.
So, I told you in discussion, we have to at least tell them what was being done. When we read this, So I want people to think this is the first non-sequential vault for cancer patient. Is it
Yeah,
true?
But as you recommended, I looked at all the articles written by Anki Barat, but
Huh?
nowhere I can find the non-sequencer. They didn't describe it like that. Even in Lancet or JAMA paper, they just said it like bilateral lung transplantation assisted with cardiopulmonary bypass. So
Hmm.
that's the reason why I deleted the word non-sequencer. But if
Hmm.
if there's any way we can confirm it by Ankit Bharat again, maybe it can be done to be oral, and if he's okay to use that word for describing his surgery, because in some other papers, I can definitely find the word non-sequencer or sequencer, describing the transplantation done for this kind of patients. So I think it's okay to use that word, but
So I know for a fact the both lungs were out simultaneously. So I think it's fine to use non-sequential.
Okay.
Yeah. And in other lung cancer, lung transplant papers, did you find any article that talked about non-sequential or assisted by cardiopulmonary bypass or ECMO? Did you find any of the articles?
Up until now, no. They only did sequential lung transplantation.
Yeah. So I think you should say this is the first one ever tried. The first approach.
The first, okay. May I limit the patient to the invasive mucinous adenocarcinoma?
For this case, it's one case, so you can say that. Well, no, I think for lung cancer, lung transplant history, this is the first one.
Okay, then I'll double check if that's correct. Okay.
Yeah. So again, in the discussion, we have to say what has been done in the literature. Literature is saying they were only a few case reports.
Yeah.
And I think you should cite the NEJM paper.
Oh yeah,
I think you should.
Yeah.
And then I think you should, because we're not claiming we're doing transplant for the first time for lung cancer. It's been done, but it's been done with very limited case, and it was I guess in sequential manner. This was the first non-sequential. Also this, and then I don't, did any of the lung transplant case show MRD?
No.
So also you can say this is the first lung cancer, lung transplant case that had serial MRD assays done as an association or as a translation medicine study or something.
Yeah, totally agree with you. May I?
So you have to tell them this is the first one. I think that's very important.
Okay, so that's the reason why I modified our title as well a little bit. Could you look at the title? So I put surveillance utilizing minimal residual disease assays. Do you think it's worthy to be noted also in the title as in itself, or? So title used to be just bilateral orthotopic lung transplantation for a patient with advanced IMA.
Yeah, I think you can still put, so the people sometimes would search title by, like you can still put these.
Gotcha.
Oh, you forgot the big one, Eugene, of lung.
Oh,
You could say pancreas here. You know what I mean? You can say,
the most important word. Yeah.
yeah.
Okay, thank you.
Yeah.
Yep, then I will modify it. Reflecting
So
all
it's
three
same here of lung.
of long. Oh, okay. Yeah, I'll make sure there will be long for every.
Previously called, previously. To where's? Okay. Yeah, so plus medicine, so look at their guidelines. So I think first this shouldn't be table 1 because we're, MRD kind of comes as a later translational medicine.
True.
So all the numbers has to be in the order it appear in the text.
Okay.
So I think this should be,
But that's the figure, so we only have figure 1 and table 1. So.
oh, that's why it's table 1. Okay.
Yeah, is there anything we can make table? Like, I'm not sure. Yeah.
Well, he doesn't have PET scan. Does he have any PET scan before?
Can anybody look it up? I'll look it up.
So if he has PET scan, I don't think we did PET scan post surgery. But if he has PET scan, I think you can put that as a supplemental figure, like representative images to show how, where his disease is.
Yeah, only limited in lung. Yeah.
So you can show that. Oh, so
how about summarizing all the MRD assays result in one? As of now, we have Signatera result in the figure and Guardant result in table 1. If I find Tempest result and then can I summarize that also as a form of figure?
so table 1 can be, table 1A could be liquid TTDNA profiling, and then it could be and then the next table 2, I guess table 1A, 1B could be, and then yeah, it's not quite a figure, but table 1A, 1B could be tissue, DNA profiling, Tempus, and you can make a table out of the mutations you found. And I don't remember in the garden if 4 were all functional, if there was any VUSs.
Oh, there was. So map K3, they noted it as VUS actually.
And other 3 were functional?
No, other 3 there's no remarkable remarks. There's no remarks.
That means it's functional.
Oh.
Yeah. So I would make that, maybe put asterisks next to MAPK3 and then put BUS.
Mhm. For sure. Okay.
For Tempus, I think you should both report functional and BUS and then allele frequency.
Mhm.
In
a table.
and
right?
yeah, and then if Guardant, any 5 of this, any of the reported TMB, you should
TMB was not detectable, non-detectable.
non-evaluable
Sorry, correct, non-evaluable.
in all 5 reports.
Do
If
they
you have,
each TMB for each variant? I didn't know that. They only, as of now, I can only see the TMB in the front page. But
no, I'm saying there's 5 reports at 5 different time points.
oh. Yeah, let me look it up later. Here's no.
Yeah.
Yeah, okay.
So if it's non-evaluable, I would just say in the results that all 5 time points TMB was not evaluable. Blood TMB was not evaluable. And I think in the tissue it should be evaluable, so you could write that.
Right.
The
Tissue
table.
was positive. Yeah. TMB was initially 1.
Yeah. And so it's important to know what's overlapping. So you could, in the results, say, let's say Tempus tissue found the exact same TP53R248Q. If it wasn't exact same variant, it's not important.
Okay. But
if it's same, yeah, if it's same, and if it's detected even after 3-6 months after surgery, then that by definition is MRD positive disease, which Signatera clearly missed.
Oh, interesting. Gotcha. Yeah.
Okay. So that could be our table.
Discussion point. Yeah. And then table.
Yeah. 1B. Yeah, so many things to discuss actually. He has Veristrat, so a Veristrat can
1C table or.
But I don't know if he. That's just one N of 1. There's no comparison that can be made. I mean, if it's Veristrat is good, you don't have to report it. If it's poor, we know you can just say his proteomic signature was reported as poor risk, which is known to be associated with poor response to chemotherapy or immunotherapy.
Okay.
If it's good, I mean, you don't have to report it. It's
Okay.
I mean,
Understand. Okay.
And did you report lenvatinib did work?
Lenvatinib, I brought the thing Isel mentioned in her abstract. So in the case presentation part, when you look at the second paragraph, the very last part is discussing about lenvatinib. So while awaiting board, lenvatinib 20 milligram daily.
So you have to be convinced. You have to read the patient's chart and be convinced that this is true.
Okay.
You can't copy paste either.
Gotcha.
Improvement in respiratory failure. Improvement in oxygen delivery. I think that's very vague. What is improvement in oxygen delivery? Did you change the setting of the medical mechanical ventilator. So
The amount of O2 level, yeah, okay.
so the oxygen saturation improved. That makes sense. I don't know what oxygen
Mhm.
delivery means. So you in choosing terminology, you have to be very specific, like in improve from what to what. You know, oxygen requirement dropped from 10 liter to 2 liters.
Got it.
You know what I mean? Like you can't be vague about these things.
Mhm. I think that makes point, yeah.
Wait list time. Okay, I think you can. I think this is still a bold statement. We don't know this can be used. I mean, we can say, we can describe the fact. So far, 6 months, patient has no radiographic sign of
Recurrence.
recurrence. And the tissue-based assay, MRI, but you can, like, let's say if you, and I think maybe Tempus, can someone pull up the Tempus report and there's, I mean, you can do it, you know, right
Yeah.
away.
Right, I will.
Can someone do that? Then we're gonna have to say he's MRD positive. If
Mm,
none of what Tempus discovered matches what Guardant discovered, you can say what Guardant is picking up is not validated by his tissue.
yeah, still remains unclear, yeah.
But what's clear is Signatera that says tissue-based MRD is tissue-based blood MRD is negative. So you can still emphasize that.
Okay. Yeah, I'm pulling
With
up
so much unknown and so short follow-up period of 6 months, I don't think you can say can be utilized.
yeah, I agree.
So you just have to say, you know, 6 months out, he's doing well. It remains to be, you know, the further
Surveillance,
further follow-up
is warranted.
and larger registry trials are awaited or are warranted, something like that.
Actually, thank you for bringing up that point. So actually, I deleted that larger study related writing deleted because of the word limitation. But if we can write
Yeah,
out,
but you can do +1.
but yeah, in that case, do you want me to mention that as well?
No, because you're submitting to PLOS medicine, I think you should write that. It's important. And it's important to mention that we are starting a registry trial to explore this question.
Oh, even though we haven't approved yet, we haven't been approved yet.
You can say we are starting. Starting means we're writing up the protocol.
Okay,
It
got
doesn't
it.
matter.
So I just pulled up the Tempus.
Can you share?
Yes. But unfortunately, variants are not matching with Guardant, but I will share it.
The Guardant only had that 4.
Okay. Oh, can someone unshare it? It says I cannot show.
You cannot share it. Okay.
Can you see the results here?
Mm-hmm.
This is
So
done at
I think Albert Curie has two tempests. One is coming from, so you can put two tempest results. One at diagnosis from biopsy.
Mhm.
The other one is from surgical resection for transplant. And I think it's gonna be convincing if the two of the tissue NGS picks up the same variant that Guardant is picking up. Because it's blood times 5, tissue times 2. The question is there any overlapping DNA variant?
Right. So you mean we need to compare tempest with 2 time points tempest results. Um,
So you don't have the report? Tempest report is in the media file.
media file. Yeah, here there are so many media file that so
it will say Tempest.
tempest. Okay. Just give me a sec. I will.
Eugene, I think there is one in December 21.
December 21.
I think so.
Mm-hmm. Thank you. December. Yeah, here.
There's like a whole bunch though. Let me keep going down. We can look at the December ones first.
Mm-hmm. But weird. I can see Signatera and Guardant, but Tempest, I cannot pick that up.
What about Altera? Because Altera is Signatera's NGS. Click
Uh-huh.
on the Altera. That's you can click on the Altera. Go down.
Oh, here.
Yeah. Click on Altera. So low tumor mutational burden.
Uh-huh.
And there is, go to next page.
Oh,
So they found all these VUSs.
true.
So can you see any of them matching Guardant?
Sure. TP, there's no T. TTLL is not. And MEP, there's no one. There's no M and NRAS, I cannot see it. And for TSC1, yeah,
Can you go down?
okay, for sure.
Okay, so that's the only. So you could make a table for what Altera found. So just validate it. So you can put this as a supplemental figure
Mhm.
For another NGS.
Mhm.
I think this is coming from surgical tissue. You have to confirm it.
Okay.
Okay, go to Tempest,
Sorry.
close it,
Tempest.
go click on the Tempest.
There are several Tempest not RNA, right? MMHRD.
XT is the, yeah.
Yeah.
And you can report MMR, HRD, everything in the result because okay, so TMB is 2.1.
Mhm.
Go down.
Oh, here,
They found these 3,
true.
and this is coming from, is this a transplant tissue? Is it,
Oh.
he,
This was done, this was collected before surgery.
no,
Tumor
that's normal specimen.
Yeah,
When was,
it's collected on May. 4 months preoperative period time point.
that is normal tissue again. Tumor
Oh.
specimen was collected on September 25th. So when was his transplant date?
September 24th.
Okay, so this is surgical tissue. Now, go to RNA expression report.
Oh, I'm sorry, yeah.
RNA expression report.
Mhm.
Up. So you could say RNA profiling showed underexpression of this in
Mhm.
the results section. Okay.
Mhm.
And then go to Fusion. I think Fusion report.
No,
No reportable fusion was found. And then go and find another Tempest report at diagnosis. If you go down,
Tempest, yeah. Maybe around the time he came to this hospital. Fairy structure here, gardens.
okay.
I think this is the very first time he came in.
So do you have Tempest ID and password for the Tempest homepage?
I think I can know that, but I've never tried that by myself, but yeah.
So Juhi or Sungmi or anybody or Arnie, can you look up Albert Curie to see if there's any other Tempest report? I thought we had 2, but I may be mistaken.
Tempest. Tempest, the first Tempest that I'm noticing here is on 11 November. Just for PDF. Tempest requisition is done November as well.
So those are requisition forms.
Yeah.
Click on the PDL1 report,
Mhm.
then we'll know whether that was done from surgical tissue or not. So look at the date. The
Mhm.
date collection is September 25th. So this is a surgical
Oh.
tissue.
Alright.
So you should report all these tumor proportion score. Is there an immune cell score? Oh. So his PD-L1 was negative, but his combined positive score was 3, which means that there's the immune cell score is 3 because there wasn't any tumor cell.
Okay. Yeah, I can find his accession number to Tempest here. Maybe I can go to their website and then.
Yeah. So Artie, Sunmi, Jui, can you find any more Tempest report? No?
I'm doing the website thing.
Opening.
Yeah, I went to the website
Thank you.
and
Hmm.
he has 3 done.
3 Tempest done?
Yeah, let's see.
Can you share your screen then?
Okay.
I will unshare it.
I think, can Eugene do it? Do you think? Okay.
Wait,
then-
can you do that for me? Yeah.
Yeah, sorry. Let's see.
Share screen. Oh, Eugene has to undo it, I think.
Oh. Sorry.
That's okay. Yep. And can you see? Oh, okay. Wait. Yep.
Oh.
So yeah, so there's, which one would you be interested in seeing?
All of it. Well, XT.
There are 3 XT?
XT and that's
So this is coming from sample collected June 2020. So this is at diagnosis Evanston Hospital. So you can kind of compare this side to side in a table.
Mhm.
There's a copy number loss. Can you go down? Okay, so there's still same. So this is very interesting and that they're very accurate. These VUSs detected at diagnosis and at surgery, transplant surgery, are exactly same mutations. Not even, so this is pretty accurate, and none of these are detected in Garden panel, vice versa. So this gives me, and then Signatera was negative, which is another tissue-based assay. So this is two counter argument that you can make. And this attests to maybe Seungmi's article that tissue-based MRD may be more meaningful, because if you do tests like Guardant, you have no idea what's overlapping between tissue and blood. And for Albert, Guardant is becoming back, always positive. So you could argue, and then and it's two different companies using two different platforms, Altera NGS and Tempus NGS. And Tempus was even two different time points, and they overlap perfectly. So it's not a technological error. And it's still echoing. And still there's not a single clone that's overlapping. So to me, I'm very suspicious that these very low allele frequency burden is maybe coming from somewhere other than lung.
Oh.
Okay. So Eugene O, your next destination could be look under CHIP. I think Seungmi knows a lot about it. Clonal hematopoiesis of unknown significance, so there are known most frequent mutations found with CHIP. Okay. Maybe I don't know.
I'm sorry, Dr. Chia, I don't think I get it. So VUS found in who?
No, ask me, I said, about CHIP, CHIP.
CHIP.
Because they can be detected in blood and be mistaken as MRD, but they are sometimes just the bone marrow abnormalities.
Oh, okay. Yeah, maybe that can be happening here for Curie. Okay.
It may be or maybe not, or it may be.
Maybe or maybe.
So maybe Eugene O for next week. And do this with Seungmi. Both of you can look up, and Arnie can help as well. What are, this is a very important questions. So let's do this. Well, Eugene O, So Sungmi, this is relevant for your paper. So I want you to read more about CHIP and what is this prevalence prevalence and how detrimental, you know, why is it bad, or is it really bad, or is it not bad, or and how does that falsely affect CHIP? So maybe you can just brief presentation. And Arnie, you can present on the topic of, besides CHIP, what is the what other ctDNAs can you find? Okay, one example is fetal DNA. If the patient is pregnant, you can find fetus DNA in the blood. If patient has aortic aneurysm, there's a ctDNA coming from aortic aneurysm. If patient has colon polyp, which is not cancer, you can have APC mutation coming out from it. So those are the examples. What are non-cancer CTD? You can't even call it CT because it's tumor. What are the non-CF DNAs that can be mistaken for MRD? Okay, give us a landscape, an instance, or understanding of what they could be. And so that's Arnie and Sungmi can let us know. But whatever they learn, Eugene, you can incorporate that in your paper.
Okay.
Okay, I will try to read it first, Doc. Thank you.
Okay.
Yeah.
But I think we just discovered really good point that, the Guardant. So we're gonna make this argument. Tissue-based Signatera has been repeatedly negative. Guardant has been positive, but the number one, if by Charlie Swanton's paper, that if there's a perfect linear correlation between the tumor burden size and the ctDNA size, ctDNA
Frequency?
allele frequency, we don't see that change, because before and after surgery, there has to be a big difference. If it was really shedding from the lung cancer, then it's just made that. So that's an indirect evidence to suspect that it may not be coming from lung, that there was no drop. Number two is that, like I said, Altera, Tempus has not validated any targets or any mutations, whether it's VUS or not, from the tissue. So it's unclear whether they're coming from. So I would argue that these may not be true MRD representation. So deep, right? This says you have to understand everything about technology to get to this point.
Yeah. Interesting. I got it. Yeah, I'll try to make that arguments based on what we have discussed so far. And I think Albert Curie case can reveal so many things about MRD assays, which is interesting.
Oh yeah. There's just from one case you can learn some. Imagine having case like this, like 20 cases. Yeah.
That's what. Yeah.
That's why clinical case, interpreting it, and you're actually going to make changes in patient treatment or follow-up or workup plan. It's so sophisticated, and it really relies on what the physician already know, and what their philosophy is, what their scientific perspective is. It just it's very different. Okay. I think we discussed a lot from this case. Anything else we are missing? What are you launching here?
Who is sharing her screen?
I think I'm sharing my screen. I'll sorry. It's nothing.
Yeah, so for me, other than Curi's case report,
Mhm.
I studied a bit about calculating sample size for non-inferiority clinical trials.
Yes.
I think I understand 100% as of now, and I think I have to work on more. So can I share it with you, or yeah?
Yeah, maybe we discuss it later or next meeting.
Okay, got it. Then I think I'm done. Yeah.
Okay, and what about the protocols? Are we waiting or?
A protocol, Liam, can you update us?
Yeah,
Yeah.
Liam.
So we have the two protocols that are kind of in the finishing kind of stages. So for the first one, the double lung transplant protocol, Dr. Engelbart sent back both the protocol and the consent form. He sent us 2 emails, and I was asking some questions that required your input, Dr. Che. So we were waiting for you to take a look at it.
Yeah. Can you
Yeah.
share your screen? Let's go over it now.
Okay, one moment. So Dr. Che, are you coming into the office today?
No.
Okay. Give me one moment. And then regarding the second one, the biomarker studies, we sent out the emails to all the named organizations or the laboratories. They've all responded back, and they're very eager to set up a Zoom meeting with us. So I believe Leslie should be working on scheduling those for us. There was only one place we were waiting to hear back. They were traveling, so they had the automatic reply that I received. Yeah.
So all the companies we sent to, they're interested?
Yeah. Yes.
Surprisingly, yeah.
So I'll share that screen first. So just to give you an update of where we are in our communications. So this is the list of companies we reached out to. And they have all responded with the exception of Tempus. Tempus, I got the automated response saying they're out of office till the 2nd, which was yesterday. So I believe we should be hearing back from them soon. But everybody else responded, and they were eager to hop on a Zoom call to discuss the details. So yeah, very receptive and really excited to see what those meetings are going to show.
Good.
Yeah. So
maybe for the preparation for the meeting, the blood we're going to draw, and just make, and then we would not disclose any other partners that we're interested. We would just solely focus on that company and what they need, what are the samples. So maybe the bullet points to decide, how much sample do you actually need? Do you need, what kind of tube do you need? And do you need a special treatment? Some companies would ask, let's do a centrifuge. Right after you get the, it has to be shaken and it has to be stored in negative minus nitrogen tank. What do you want? And then I think we're going to advocate, we're not going to send it to them real time because this is all research. The real time knowledge would not change our management, but we would all look at this as a batch. Like after we get 100 patient samples, we will send them as a batch, and to reduce any batch effect, that's the best way to analyze it. So we would recommend that. And also, some companies will do, what is your investigator initiated trial like submission form. So they all have their own form. So we would just fill out the form and submit it. So this will be more like understanding what our mutual interest is. Are we able to work together, and what are your requirements? And once we click, then we submit the form, and they go through their own committee, and then they approve it, and then we send them budget, and then they give us money.
Hungry. My lunchtime.
Okay.
We should let him, I think we should let him go.
Wait.
내가 먹자. And then the protocol, thank you, Bharat. Thank you, Bharat. Let's go over that.
Sure.
Give me 2 minutes. Let me. All right, let's review the protocol.
Yes. So the protocol. Let's see. Do you want me to share this screen, or do you want to pull it up on your end, Dr. Che?
You can share your screen.
Okay. So this is the latest version Dr. Bharat sent over. Yeah, please excuse some of the formatting changes. I think it just might have to do with the Word versions we're using. Yeah, so that's the title.
So what change has he made?
Unfortunately, it's kind of hard to tell at this point. Yeah, the formatting that I'm seeing is difficult to tell.
By any chance, did he mention anything in email directly?
No, so I can, why don't I go over to the emails then? Let's see. So these are the emails that we need to discuss with you, Dr. Che. So these last two emails are the ones that came from Dr. Bharat. The first one is regarding the consent form, and this is mainly where we need your input on. The second email here on the bottom is regarding the protocol. He didn't say much, he just said, made some changes, okay to submit from my viewpoint if Young agrees. Ray Tomic has no further comments. So that was it for the protocol. And then for the consent form, there were two questions that we ran by Dr. Bharat. The first being the contact information, just kind of double checking, is it better to put my number in there or to leave Dr. Bharat's office number there? Just checking if for any surgical purposes or reasons they needed to have the surgical team's number there. But Dr. Bharat's pretty indifferent, says it can be whatever number. So I guess that's up to us to decide. And then the main kind of question we wanted to run by you is this part. So all these patients will be consented for the lung biorepository, and we will be able to collect the specimens that are described in it. I wonder if it would be better to keep the tissue collection as part of the biorepository consent that we have and the consent to be part of the registry as a new consent. What do you think?
I wonder if it would be better to keep the tissue collection as part of the that we have and consent to be the part of the registry as a new consent. We consent to and the consent to be part of the registry as a new consent. So let's do this. The actual consent to tissue the actual consent to tissue storage will be done through the biorepository consent form they are already using in thoracic surgery department. I think I don't know anything about their blood. I don't think they have any protocol that stores blood. This so I think for lung transplantation MRD assay, I think we are going to say I feel like all these lung transplant patients are going, I feel like they are going to be followed through MRD assay. However, these are not the exact same protocol. Like if we run out of fund, or if there's any hiccups, you know, with regulation, I think there will be issue. So I think for blood, we're gonna have to get consent through this form, and I think we will collect blood every 3 months, as we said. And I think we have to specify, you know, to say the blood will be collected in here and here and stored in aliquots in biorepository or bank, thoracic surgery biorepository or something. And then we can say, in case patient consents to a different biomarker study, then they I feel like it will be always double.
As in, they're basically going to be consented for 2 different trials.
Yeah.
Yeah.
I mean, it's consenting for blood and tissue. So
So their biorepository study, they collect blood samples too. I just don't know if that's enough for what we're trying to do as well.
no, I think they collect, I don't know. Surgery department to send you their biorepository consent form and the protocol and see.
Sorry, Dr. Che, you broke off for quite a bit there.
I think you can, I think they can consent for both. I think we, you know, I think we should say your sample from your tissue sample and blood sample will be used for our, you know, translational medicine research as part of this protocol. So I think we'll leave it there.
Okay.
Because it's just consenting to, you know, use of tissue. For that, they don't have to do anything. We're just gonna, look into the archival tissue. But for blood, for them, it's a threat. It's like poking with their skin. So I think I want blood to be drawn as part of this registry trial. And let's see if it can be done solely through, so consenting. And you can say this, it could be done through the biorepository trial or through a different MRD protocol. You can say, we could store the blood for future use, or we could, in lieu of the future research storage, a patient can also participate in biorepository protocol or minimal residual disease assessment protocol or something. We can, or you can leave it vaguely. Patient can also consent to other studies, something like that. So I think we could leave it like they can consent to both. I just want this to be more overarching and make sure even if patient misses, forgets to sign the other two protocol, then that the blood is already collected and it can be used for later future research, something like that. So
Okay.
I do think there's a language that, yes, I'm consenting to have you guys use my tissue and blood for future research. That's all. Yeah. And it is okay to sign consent for other two protocols out there. Maybe IRB would maybe ask like, whether between the two trials, you guys should not take more than 70 cc's or something like that. But I don't feel like we should put that unless IRB directs us to write it in.
Okay. So, but the bottom line is we're going to still keep the tissue and the blood sampling in our protocol and consent.
Right. But also say this is just overarching statement that you consent that it's kind of consenting that we can use your tissue and blood for the purpose of this transplant registration study. So it's consenting to the purpose of your sample use, but the actual sample can be collected through other protocols as well. But if they haven't consented to those studies, then we can use this protocol to really get the sample and store it.
Okay.
So it's
so
a safety net.
yeah,
It's a
yeah,
safety
yeah.
net. Understand there's a safety net.
Okay. So I guess we're not changing much about the language then.
Yeah, but I want you to read the biorepository and see how many times they require blood draw and sort of see if we can use if they're doing it every 3 months, great, that's what we want. If they're not doing it every 3 months, then I really want them to consent to every 3 months and then store it under our protocol.
Okay.
You understand? Yeah.
Yeah. So we're what we need is every 3 months.
Right, and they might be collecting 20 cc's. I want 70 cc's for future research.
Okay. So if they are doing 20 cc's, do we need 70 independently? So a total of 90?
No, it has to be 70. And even in our MRD protocol, which is separate from transplant protocol,
Yeah.
I want at least 10 cc for future research. We can't send out, like use up all the samples.
Right.
So the future research has to be 10 to 20. And I think that's what the thoracic surgery biorepository is.
Okay.
And when you ask them about the protocol and consent form, ask them about their inventory, like how much they have collected and did they actually look at the longitudinal? That way we can talk to the Enki Parad about using those samples for our MRD studies because they already have samples collected and patient outcome is already there.
Okay.
So we can do retrospective and then prospective, both for MRD assays. My guess is they just get a blood, pre and post surgery, and that's it.
Mhm.
If that's the case, then the blood has to be solely dependent on the transplant registry, MRD. And the ideal scenario is all lung transplant blood should be analyzed through our MRD protocol. Just like Albert Curie was in our initial 100 patient study that was already done in U1701. So
Okay.
that way all different company platform can be used.
Okay.
So just come up with a language that covers everything we just discussed.
Okay.
Yeah.
Then we'll make those changes. So just to clarify, regarding the volume of blood we're drawing, we want 70. And if the biorepository is already drawing 20, do you want a total of 70 for both, or does that number have to go up?
So I think biorepository. So let's look at what they say first and then decide. Yeah. So ideally, whatever we wrote in our MRD protocol is what I exactly want to do for all transplant patients. Okay, pre-surgery, 4 weeks after surgery, and every 3 months, right? So, and then that's exactly what I want to do, but I don't know what biorepository protocol, it says. So let's compare and see what we can do as a, medium.
Okay. Yeah, I have that information in the lab. I should be able to look it up pretty easily.
Okay.
Yeah. So yeah, I'll get that information and I'll reach out to you, in the afternoon.
Okay.
Yeah. So if we have any questions throughout the day, should we call you?
Yeah, just text me, then I'll be able to call you. Yeah, I'm still available.
Okay. All right. I think that's it for now. Do you have any other comments, Dr. Che, regarding the protocols?
No, not at the moment.
Okay. So
once
with
we
regards
fix it,
to, yeah.
So still navigate IRB. So I don't know if IRB or the lung cancer protocol coordinator, I don't know if she responded. So I CC'd you guys. So the question was, can this be tagged along with the existing NU1701?
Oh, the one you sent to Brooke, right?
Yeah, did she respond?
Brooke responded. Let's see. Yeah, so Brooke said, thanks for sending this update along. This should go to both the IRB and SRC for review and approval. On the SRC side of things, do you mind sending over the revised protocol along with the summary of changes? We can submit this for SRC review once we have that documentation. Please let me know if you have any other questions.
Yeah, let's do that. So once we have the final draft, it's going to go to send it to Brooke, and she'll send to SRC, and she'll send to IRB. So she'll be the navigator.
Okay, so the final draft, is that the version we make after we discuss with all of the collaborating labs?
Yes.
Okay. So once we have all of that done, we'll send that to Brooke once that's ready.
And we don't have to submit the appendix to SRC or IRB. It could be the participating labs. In the introduction, the labs will be analyzed in, I don't know. Yeah, it could be still vague, and I think we could still get approval that way. So we
Okay.
don't have to say which labs are participating in what.
So in that case, I guess we can just organize the summary of changes and then send it to Brooke, right?
Yeah.
Okay, then we can get started on that. Just one more question regarding the communication piece with the collaborating labs. So I assume Leslie's, in communication with them to set up appointments. Is there anything like we need to follow? Like, do we need to follow up with Leslie, or does she know to just automatically kind of put that in your schedule?
I'll just email Leslie when they invite participant. Make sure they invite you and Eugene. Oh.
Okay.
So that it's in your calendar too.
Understood.
Just CC me, and yeah, so that it's not missed. If I am only there and I miss that email, then Okay. So let's finalize a general form and let's submit that to IRB and SRC. So that way we save time and get that still approved by IRB, SRC while we're talking to companies.
Okay.
And once we finalize it, you can send it to Brooke. You can say you're the clinical. So the formal title is clinical research associate, working with me and Dr. Bharat, and say you wrote this protocol with me and you're going to submit to SRC and IRB, or have Brooke submitted, and then also navigate, like how do you add your name to IRB? How can you consent when consent patient, like what's the pre-requirement, and is there what's the training, and how do you navigate the IRB submission page? So learn all that skills while we're going through all this.
Yeah, sure.
Okay, anything else?
Also, may I ask a question about transfusion transplantation protocol?
Yeah.
Yeah, based on what we've discussed in the past, and then regarding on based on the previous articles that I reviewed, I'm gonna make a brief, just 1 or 2 pages proposal. But do you still want us to work on protocol itself, or is it okay just as of now? Is it okay to just prepare that 1 or 2 page protocol like proposals other than protocol itself?
Well, we have to make that into a protocol. So we have a proposal to companies, but I think we have to start making it into a protocol, just like the MRD protocol.
Oh, okay.
So it has introduction and who are the patients and how, sample number and what are we collecting and how are we collecting. So I think we talked about transplant and transfusion. I still don't know how to do it in the US. So, is it Red Cross or is it our blood donation center?
Yeah, honestly, we haven't looked that kind of information yet. So,
and maybe we start the conversation with Marcelo, email him, send him our our protocol or our proposal. Say that we're in talks with Cell Search and ctDNA companies like Tempus or Guardant or other companies, you know, because Marcelo knows everything. So tell him this is what we're doing, and tell him that we are, once we you know, decide on the partners, this is our plan to draw blood before and after transfusion and follow them up. So what is feasible in Brazil? Sao Paulo, the blood transfusion center, because they have to have their own protocol. They have to go through their own IRB. And once they get the blood, it has to be sent to US. There's a lot of shipping going on to that lab. You know, we need to discuss what is your volume? Like what, you know, do you think, you know, we could consent this much of patients? And what's, you know, can we do this in, you know, 200 patients, you know, what are the expected volume over, let's say 1 year, and how, you know, and what are they in an environment where follow-up is even feasible? Like, is it like one time, you know, are they all in the same care system where we can track whether they develop cancer in the future? So a lot of like these practicality questions. So maybe work with Marcelo, Dr. Cruz. Hey, we're kind of giving him updates. This is where we are. We're writing protocol. If we really want you to be our part of the transplant, it will be here. But the transfusion, I think you guys have the bigger center, maybe easier to do mass blood draw and follow up. So, yeah.
Okay.
And then CC me and let's see what he says. And I don't know how the transfusion, because we transfuse a lot of cancer patients, but we have to reach out to general population. So maybe do some brainstorming and read some of the transfusion research articles where, you know, what is the best way to do it? Maybe you can call Red Cross, or we have a blood transfusion center, but we're not. We need maybe we have to think about, because we did a first delivery woman, right? So we have to think about a specific group. But if we make it too specific, we're not going to get there. So, I mean, we exclude cancer patient, and then what, you know, cardiac patients fine, but I think we have to give it more thought. What's our best population, and what's feasible? You know, what's feasible here, what's feasible in Brazil. So really think hard. This is a good question, and how to prove it in real world is really challenging. But
Yeah, we'll do some more research on that.
yeah. So Liam, you're part of this. So brainstorm with Eugene. You know, what is U.S. transfusion system, and how can we get them consented, and they all might have different insurance, different PCP. Like, how do we track whether they have cancer or not? Is someone gonna call them every 3 months to see, do you have cancer now? You know, like what's the best way to track their cancer incidence? And what are we trying to provide them? Like, I was actually thinking, like, if we have some sort of deal with Gallery, and then we can screen them for cancer, you know, and then provide them the report with the Gallery report. And it's a research, but it's also giving them a free option. And it's really up to them, like after discovering, oh, I might have cancer in my body, and then you might initiate looking for cancer in their body, right? But it's still done part of research. So this report will be available after they finish organ transplant or after they finish. And then we have to think about what's ethical. But I think if we say this is because knowing they have cancer risk, can they still donate blood and donate organs? So that's a whole nother thing. So we could, but this is how germline DNA researches are done. So we can collect, and like we said, we collect 100 patients, then we can send them as a batch and run it the identified manner or something. That way, there's no ethical issue or something like that. Or we can make it, we will provide the risk, and it's up to you to do further workup. And we've done our due diligence in detecting any cancer through our routine radiographic and all the tumor markers. Galleri is not FDA endorsed. It's still part of research. You can buy it commercially, but that doesn't mean it's medically endorsed by, so you can kind of view it that way and get funding to buy Galleri tests for our patients through funding and then do it. So, we can think about different ways to do this screening. So this is really epidemiology. So Liam, you come from MPH category, so think about it. This is public health issue. So I think let's give it more thought and think about it for a couple more days and finalize how are we. Transplant's easy. We're a transplant center. We're gonna get blood drawn for all our lung transplant patients. But for transfusion, at what center, how many people, what population, how are we gonna track them, and and then the results of cancer risk. How are we going to convey it to them? Are we going to just de-identify, keep it confidential? And either one, as long as they know what's going to be done and if they consent to that risk, it's fine in either case. Nothing is unethical as long as you disclose and they are okay with the procedure. So yeah, let's think a little bit more about the structure of the transfusion study.
And do you think it might be helpful to kind of bring up this discussion at the end of our meetings with collaborating labs, just to kind of get an idea of what their thoughts are? Or
you mean blood transfusion, about blood transfusion?
Yeah, like you mentioned Grail. So
yeah, no, we had a discussion before.
We did
but,
talk on this topic.
But yeah, not many were interested, and they don't have funds to support our study. But their intent is to sell their product. They say, oh, sounds great, use our product. So it's a biased discussion, whatever we do. Yeah. Anything else?
No, I think we're good.
Okay. All right. So yeah, just text me or for any further questions and we'll discuss further tomorrow. Okay.
Sounds good.
All right. See you guys. Oh, wait.
Actually, Seungmi.
Oh yeah, Seungmi didn't present.
Yeah.
Sorry about that.
That was funny.
Okay. Let me go and check on kids. Give me a couple minutes. I'll be right back.
Okay. Mine is short.
Still raining out there. Okay, Seungmi, you can present.
Yep. So I don't have much to present, but I think best way to start is with the manuscript.
Mhm.
So with this feedback, I could see how wait, I'm getting an echo, so let me just connect close to. I just want to get feedback, I guess, with my,
So what is your timeline, Seungmi?
I leave at the 14th.
Uh-huh.
And I should have done all the manuscript, including the discussion by today, but I haven't really finished all of that. And then it was by next week, I was supposed to have everything ready and then submit it in the following week. And then it's that would be my last week.
So another thing is have the Signatera team review our manuscript.
Okay.
You know,
Echo for some reason.
we wrote. They won't be co-authors, but I think they can still read our paper and give us input, or yeah.
Got it. Should I share you the link?
What link? Oh, okay, yeah.
Sure.
Yeah,
I think
send me
that's
the link. That's better.
Oh, there it is.
So unshare me.
Jimmy, can you share it? Because you own the manuscript. Oh, unshare. Oh, sorry. Yep.
Okay, do you guys see it?
Yes, we can.
Can everybody mute, please? I think Arnie and Eugene's other accounts.
I think is Dr. Che. Does he have 2 accounts? Because I think it's from Dr. Che, is it?
No, I have.
Oh.
Who is Yang Guangche?
That was Juhi.
Okay. ctDNA level monitoring?
Okay, that's the abstract. Can you go to the can you go to like the what page? 6th page?
This one?
Nope, the one below. This is all the abstract part. The one below. Yep. And the next one. Yeah.
So what is the title?
I'll have to work on it. Oh,
This one?
I think so. I didn't work on finalizing the title.
The title is important.
Okay.
And you need to have abstract for the paper too.
Oh, okay.
You can't just start with, so So another writing class 101. Start with abstract. Okay. So you need to know what are the key sentences, and then always start with a title. And your way of reflecting whether this is true, like really good description representative of the topic, is always going back to the title. Is a title accurate? That's number one. Number two, am I only discussing the title? If you discuss something, spend some time, like the lung transplant paper talks about, why we gave EZH2 inhibitor. That's wrong, right? So if it talks about why ctDNA MRD assay is a really good way to track disease, it's wrong. You just say it's given. It's you don't spend time if it's not related to the title. Spend a lot of time if it's related to the title. Okay.
Okay.
So, and then try to use acronym, because people will look for acronyms to find their article, the article of their interest. Or ctDNA, or you can do ctDNA MRD assay. Maybe that's too long. Let's just leave it there. No, just leave it there. ctDNA. Maybe this is better. ctDNA MRD assay. So my concern is that this is kind of a Me Too paper. So what is so unique about this paper that other papers have not proven? So this is a tissue-based, right?
Mm-hmm.
Tissue-based assay predicts outcome in lung cancer patient. You can say real world data, or some people say real world data. So Seungmi, is this paper first lung cancer paper using Signatera in clinic?
I think so. I think so, but I used the word Signatera to search for it. Would I be missing anything with using Signatera if I use the term Signatera to search?
I think searching is not the best way. I would email Signatera people. Because they would know better than anyone else.
Right. Got it.
So can we say it predicts, or is it seemingly not sure? Are you sure you can say predict?
There was definitely a statistical significant with overall survival. Despite there being like really few patients, there was like the Kaplan-Meier. Oh, I guess it's only the Kaplan-Meier curve, so we can't say predict for that. It's not logistic regression. So.
No, it doesn't matter which one, as long as you think there is any meaningful difference in at least one measure, it's fine.
Yeah, extracranial, I think, yes.
Extracranial.
And overall, overall, yes.
Provide predict survival outcome in lung cancer patients. So do we have any small cell, or is it all non-small cell?
We have 2 cases of small cell.
Okay, lung cancer patients. who had curative patients who they had. Curative treatments. Curative treatments. Curative treatments. I want to just convey that message that this is the first, but this is the real world data. This is not like a clinical trial or tissue-based predicts outcome in lung cancer patients. Oh, so this MRD assay, post-surgery or post-chemo radiation. Well, let's leave it as is. Let's think about it if there's any better title.
Right.
So are these, so what do you want to achieve this? Like, you want to ask questions or?
I think if you could have like, give me any feedback with the introduction first, and then we could start from there. Because I'm not too confident with my way of writing, and if I get feedback, it would help me, write when I finish the other parts.
So here you talk about sensitive, specific, and then you talk about significance.
Yeah.
And then you talk about advantage.
Mm-hmm. It's not a good flow.
And you talk about ease of plasma sampling.
Mm-hmm.
And you talk about MRD. It's all over. There's no one theme.
Right.
And then I go to second paragraph. Utilize with some level of clinical confidence. That's a vague term. What is clinical confidence?
Yeah.
Predict the recurrence affirmed by high positive predictive value. So what is tissue-based MRD has found most widespread? Major clinical guidelines such as NCCN are yet to endorse. So when you say previous studies, what studies do you have in mind?
There's the one done by I think there's 3, and none of them has small cell lung cancer. 3 that are similar to ours, like retrospective, looking at patient data, and then I don't think any has stage 4 because they were all with curative intent, and some were like limited stage 1 and 2, and some had stage 3. But I don't think we had like a single stage 4 in any of those. So that was something that was different. And then, yeah.
Mm. I don't think that's a selling point of this article. I think that's something that can be discussed as a, something that's different from other articles in the discussion. So you should focus on stage 1 to 3. For MRD study, you should focus mainly on one histology, which is non-small cell. We're shorter in number, so we added some small cell and to understand what small cell is doing, but it is not the selling point. If we were really interested in small cell biology, we would have done, small cell or focus study, or at least it's half and half, but it's just a minority of patients in our study, small cell. So I don't think it's a selling point. So writing class 101, one idea in one paragraph. Okay. So ctDNA based MRD assay is a new concept. What is it? And how is it being applied right now? So and why is it important? So that, even before you discuss this, you should not go into what is the sensitivity, specificity, and what is the feasibility. You can't talk about accessibility, all of those in one paragraph. You have to define what it is, definition, and about the technology, why it's unique, and so importance, concept and importance is one paragraph. And then the current status, like how is it actually being applied in clinic? How is it endorsed in the clinical practice? What is the paradigm in other tumors? Now, what about lung cancer? So that's another paragraph, or you can combine it with what's happening in lung cancer is really not ideal. So there's a gap of knowledge. Like we're not using this as much as we should. And if there's 0 study, you should say there's no study looked at it, reported yet. If there's 1 study, you would just say, there's 1 or 2 studies, is a limited study have been done, very premature or something, and this is this is a first report of its kind, looking at and you can briefly mention that in the MRD there are tissue-based approach and non-tissue-based approach. And tissue-based approach is really a personalized approach where the unique 16 genes was a certain set number of gene mutations are selected. Because that's a concept, because that's our title. People may not know what tissue-based ctDNA assay is. So you have to, so this is the who's not even interested in oncology should read and understand what are they talking about? Tissue-based ctDNA MRD assay. What is MRD? Give them definition of the MRD. What is minimum residual? What does that mean? I don't even get the concept. So you have to teach this to a medical student level. So that is your introduction. So I told you guys already. Writing introduction, concept and importance, and the current landscape. And then you go to gap in knowledge, unmet need, and then you do, what did I do to address it? So you have to have that logic flow. You can't touch, you can.
Sorry.
So with that logic, you have to show that, why is our study unique? So uniqueness of our study, I mean, that's to be elaborated in discussion, but in the introduction is I don't know what this is. You say this is the first study, or this is the real world data, or and I know this may be too late, but if the other thing you could do is if any of the patients that are on this study have gardened at similar time point, just like Albert Curie, you can compare. And I don't think people have been ordering Signatera and Guardant at the same time.
Right.
So that's something that has never been done for sure. And that's what will make your paper stand out versus Me Too paper. Like, oh, like this, we already know MRD is important. Like, it's just a single center institution that's just gonna be a Me Too paper. Or but you could, just like our Curie case, like, oh, no other transplant has looked at MRD. So you have to make sure your selling point is in the title, is in the introduction, like why did we do this, so I'm sure, I mean, I don't think it's a lot. I think it's going to be just handful, just small number that will have Guardant, because Guardant is not able to be ordered in stage 1 or 2. So it's only going to be stage 3 patients. And you can do the exact same validation that I did with Dr. Albert Curie's case. Look at the tissue variant. Look at the Guardant variant. If anything is positive in the Guardant, see if it's anything that was in the tissue before.
Got it. Let's see. So actually what I did was, when I pulled the patient data onto Excel, I wrote the dates of the Altera, Guardant, and Tempus, and their TMB and the mutations. I do have the data ready.
Yeah. So why don't you?
It's like a lot. It's a lot though.
Yeah. Why don't you look at those data first? It's going to be just stage 3, which is not a lot. So. Hey, give me a couple minutes. Give me a couple minutes. Let me help Emily and right back.
Got it. Yeah, so actually I was I just pasted the mind map you gave. Richard wrote this. And like, this is the mind map you gave last time, and I thought I was following it, but like it seemed maybe like really, it was my way of kind of following it, because I mentioned what MRD is, and then I talk about the current gap here. I thought I followed it, but maybe the flow is really bad and there's unnecessary parts around.
So maybe whoever your co-authors are, have them read it and revise it together before you show me. Okay, we'll do that. Do you think this would be doesn't it sound like an abstract kind of writing? Could I salvage this for abstract? Some of these? Maybe what do you mean abstract? Because with abstract, people tend to be like more, I don't know, more concise. And then like every sentence doesn't have to flow, because they try to compact the content into like a paragraph. What do you mean abstract? Abstract for the actual manuscript?
Yeah. The parts of the introduction.
So abstract is really condensed, really succinct. So it's like you don't to a point where you don't really have to explain what ctDNA MRD assay is.
Right. Okay.
Yeah, abstract is really just getting to the core right away, and this is what we found, and this is why this is important, and yeah. So
Yep.
yeah, abstract is totally different thing.
Okay, got it. I guess we could go to the discussion and look at what I wrote. Just like it's like 2 paragraphs, but it's something I also try to follow the mind map you gave us last time, but I'm worried that the flow won't be as fluid. I do have a question at this point.
Okay.
If you look at the paragraph from the discussion, the
Okay.
one that says Signatura, which I highlighted. Signatura is they say they could increase sensitivity because it's a tumor primed approach to 0.01 VAF. But could I use what's in their website and like, of course they quoted, they use like stuff to back it up, but it's like, I feel like it sounds exaggerated a little bit. Cause like with the tumor naive approaches, once it goes below 0.5%, the sensitivity falls. But because this is a tumor, I guess, informed approach, they say that with Signatera, they could sense up to 0.01. Yeah. But I was thinking, could I use it or not?
Good question. So gardens, if you look at what we just saw this morning,
Mm-hmm.
the MAPK mutation,
Yeah.
wasn't it 0.01 or something, or was it TSC? Eugene O, do you remember?
So for garden,
Yeah. Do you
Garden-
remember their VAF?
yeah, VAF was expressed as 0.2%.
Mm.
What is the lowest they ever reported in Albert Curie's report?
0.01.
Yeah, so I've never seen that. That was the first time they reported 0.01%, which is a counterexample to Signatera's argument that non-tissue-based approach like Guardant can report variant detection at the level of 0.01%. Correct? You just saw Albert Curie's report.
Yep.
So you don't need tissue-based approach to get to 0.01%. Signatera might argue against it, but I think 2 companies are saying 2 different things. Anyway, what you can do, so first of all, you cannot ever cite or copy paste what's in the website. You have to rephrase it.
Oh no, it's not copy and pasted, just the number. This is all our words.
So you cannot cite the number and reference the web article. You have to reference the original research. So
Right.
if they have a paper that published that, you can rephrase and cite that number in the article. That's fine, because that's a published article.
Mhm.
Does that make sense?
Yeah, it is. Yeah, so chances are I won't find the article. Mm,
And if you can't find the article, you can always email them. Hey,
right.
in the company homepage, I found this. Do you have a reference for that? And I you know, I think they better have a reference, because they can fabricate things, right? So they
Right.
should be, yeah. So yeah, it's okay to cite and rephrase. Just don't copy paste what's written.
None of the thing I
Sentence.
showed you is copy and paste. Everything is rephrased. Yeah,
Okay.
no, yeah. Apart from that one, this is copy and paste. This is just copy and paste.
So again,
Uh-huh.
ctDNA, cancer detection, therapeutic monitoring, those are more for introduction. Like, ctDNA is being used for these, but for MRD, its utility has been repeatedly validated in recent studies, and cite those. So it's more like a landscape, and in discussion we're only going to talk about MRD, so you don't have to talk about therapeutic monitoring or anything like that. So again, going back to like the title, talk about the title only. Introduction is funneling, like, oh, what is MRD, what is ctDNA, and it's kind of, and why is this topic important? When you get to that title, then you only talk about the title.
Right.
You understand? That's the logic. Okay, any other questions?
No.
Do you have, did you
Mhm.
finalize table and figures?
No, not yet.
Okay, is this the final figure, or are you still working on it?
It will look like this, but I think we're gonna add one more patient.
Yeah, we have to run it again.
Okay. This is a really, really good paper.
Yeah.
This brings me to a nice segue. So when we have a Natera, we're gonna have, and Seungmi, you're welcome to join that meeting as well. So Liam and Eugene, when we have Natera Zoom meeting, I think our agenda has to be one more. Remember the PowerPoints we created for MRD lung clinical trial? We're gonna present that clinical trial, but we have to finalize on the number. Eugene?
Yeah, I'm hearing you.
Yeah. Do you have any idea of how large the trial should be?
To calculate the number, there are a few parameters that should be there.
Yeah.
For the equation, if we select the non-inferiority margin and then you need some predicted or previous, it could be previous study results, but we have some estimated results before calculating. So if we can gather that data, I think we can
So
calculate.
it's you can find it in the review article. Like what is the recurrence risk
True.
undergoing standard chemotherapy?
And you're talking about overall survivor or progression-free survivor, or you mentioned recurrence, so maybe I can use progression-free survivor, right?
So recurrence-free survival, the same thing.
Recurrence-free survivor. Okay. Yeah, if I got that number in review, I think I can calculate the number for.
So you saw what's the mermaid study, and just follow their footstep. Mermaid study I showed you.
Yeah, and in terms of non-inferiority margin, the guideline recommends us to use margin that's been previously used in similar patient groups. And as far as I searched right now, it was between 15 to 20% in between for lung cancer patients. So yeah, maybe I can use the number between that percent. And I'll definitely have a look at the mermaid trial.
Suction, or what's the endpoint?
Sorry, endpoint for.
So 15% of what?
If it's gonna be a non-inferiority trial, the margin, non-inferiority margin.
Of the primary endpoint?
Yep.
Okay. So the best way to do this is learn from prior trials or prospective trials that are ongoing. So let's look into that more.
Okay.
And even if we don't have the sample number, I'm hoping we will have one soon, but I feel like we could present the concept to Natera and see how, what they say. And we can talk about how we're going to get the funding.
Okay. Can I ask one question? So for the project you're gonna discuss with Natera, it's gonna be landscape study, or does it have any specific primary endpoints or secondary endpoints?
It will be like mermaid study. Everything will be exactly same. So it's randomization. So it has to show that omitting chemotherapy or adding chemotherapy at later time does not negatively affect the primary outcome. Then we can safely omit chemo or delay chemo to later time. And that is the goal we're trying to achieve.
Okay. Yeah.
So it's a practice changing study.
Okay. Yeah, I'll try my best to calculate the number within a few days.
Yeah, so work with Liam. You guys, this is a project,
For sure. Yeah, of course. Yeah.
Yeah. So this is epidemiology, clinical trial statistics 101 as well. This is like what is the sample size needed to run the trial?
For sure. Yeah,
Okay. Going back to Seungmi. So I think you need to finalize what figure and what, like, I know you get the new figures, but I guess, just think what would be the best way to present persistent MRD positives or converters, or I know you have the extracranial versus cranial and panel like A, B, C, D, E, F. You know, what's the figure one gonna look like? I'll leave it up to you after looking at the final data. And then whatever graph you generate, TFSOS, I think they should all be presented. If it's something that's more not the focus of the discussion, then you can make that supplemental figures with supplemental tables. The other pieces, the level of MRD, because we didn't analyze that. When MRD is detected, you know, what is the level? Or you could do a scatter plot or box plot, kind of, you know, because when it's detected, what is the level that it's detected? And we can kind of just describe that in the graph and in text as well. I'm just thinking what are there any other tables that we can so table
Swimmer.
1
Mhm.
swimmer.
Yeah, for the abstract, we had like the swimmer plot that I made with like drawing program, because and then it was a good way to
Yeah,
make
this
the-
one this one.
So
Yeah.
I couldn't find a good program to make the swimmer plot, so I'll have to go with something like this again, which is kind of like drawing using Excel. Yeah, I couldn't find like a good program that I could use.
But this looks good still.
Yeah.
And I know this is a graphic representation, but for these patients, I feel like you need a demographic table, like what is the age range in sex. Yeah. So you need and then that should include everything you got here as well, like the chemo, smoking. So just as with any clinical trial paper, you should, you know, what are the patient group characteristics, pathologic, demographic. Yeah. So that should be your table 1. Figure, this is nice figure. And then you have figure 2 is your outcome here. And you can just make a figure 2, A, B, C, D, E, F, G, or something like that. And then maybe as a supplemental figure, you can do, you know, box plot or the dot plots of showing the distribution of MRD positive values. Okay. Anything else about figure or tables?
No. Yeah. Nope.
Okay. All right, Cindy, any other questions?
Yeah. So if we go back to the introduction, there is this one sentence that I highlighted. Tissue-based. So I think we'll have to work on it. Oh, it's like the below one. Yeah. Yeah. So with some level of clinical competence, I thought like only a clinician with experience could word that. So I thought like I want to say that in real life, like it's used and people use it to make judgment, even when it's not endorsed by guidelines. So I was wondering, well, maybe we could look at it later when I have the better version, but that was something I found difficult wording.
Right. So scientific writing is something that's different. Because this still vague. What is some level of clinical confidence? So scientific writing is all about A is associated B, B is associated C. Like this, it's reported that this is this interacts with that, and it's either you use it or not. You know, currently, ctDNA MRD assays is being used in oncology clinic to assess the risk of recurrence for patients undergoing adjuvant chemotherapy for stage 2 or 3 colorectal cancer. I mean, that's a reality. A lot of people are using it. However, and then you can say current NCCN guideline do not endorse the use of such assays. You know, based something, and you can use that, and but how, and then you can say, you know, in contrast, lung space, MRD assay has, you know, has shown to be associated with clinical recurrence risk and survival outcome. However, it is not developed for clinical use yet, let alone endorsed by guideline. So that's the knowledge gap. So what I'm saying is nothing has to be left vague. It's either you do it, it's either you're being used or not used, endorsed, not endorsed. So just if it's limited studies, if it's major guidelines, just don't make it vague. It's the NCCN. And if you haven't looked up other guidelines, don't even mention it. You know, it just can't be, oh, you know, many are doing this, you know, just make it a factual statement.
Got it.
Okay.
Yep.
So writing's not easy. Writing's not easy. You have to just adapt to scientific writing.
Yeah. Thank you.
You're welcome. So again, when you present, I want you to present after all the co-authors have reviewed it. And if anything is wrong, I'll blame all 4, you know, co-authors. We're not picking it up. Okay.
Oh no. Great.
So
Okay.
Anything else?
No.
Okay.
Dr. Chen, since we're still here, I can go over the details we discussed earlier.
Yeah. This is gonna be quick. Yeah.
Seungmi, can you unshare your screen, please? Seungmi, can you unshare your screen, please?
Oh, it's not me.
Oh,
I
it's
think it's
not
Dr. Say. Yeah.
Oh, sorry.
Oh, okay. Maybe. Okay.
So I was able to pull up the biorepository protocol that they're using. And so here they are drawing 43 mLs of blood at the time of the transplant, and then 20 mLs every time. And the wording they use here is, you know, 20 mLs at the time of standard of care blood draw during each of your standard of care visits.
Each of your standard care visit.
Yeah, so it's kind of vague. So it sounds like they're just kind of leaving that up to, you know, whenever their appointments are, and kind of the middle. So when they do their lavages, this one they specify at which months they perform them, which is 1, 3, 6, 9, 12, 18, and 24. And then they also say, you know, they may collect the epithelial brushings at the time of the bronchoscopies. So this is their wording. And this is how much blood they're drawing.
43. Each of your standard care visits. They don't specify how many times.
It didn't. That's all they said.
Wow, that's wild. It could be indefinite. So clearly they just say 43 cc. So which we're gonna say 70 cc. So in your consent form, I think they like the language of tablespoon.
Yeah.
So you could do the same thing.
But then we want it to be 70 mLs for each time we're doing a blood draw.
Yeah, each visit, no more, no less, no more. It's no less. The interval of approximately every 3 months or something. We can say that.
Okay.
I mean, we can leave it vague, but I think we could. It's so the bottom line is that because they said only 43, I think we need our consent to say 70, and we'll say every clinic visit, and you know, in most cases it will be approximately every 12 weeks or more.
Okay.
Try your best to make it vague as possible for these.
Sure. And then
we're not violating anything.
Yeah. Okay. So I'll change the numbers to 70, and then we'll.
What about tissue? What about tissue language? Just tissue standard. Obtain tissue from biopsy. Biopsy tissue. Additional biopsy may be taken. Resected and typically allow us look at your medical record. Yeah, there's a lot of standard language you can adapt.
Yeah. So I brought most of this over. So I'll just change the 70 ml volume for the blood draw, then put in the approximately 12 weeks language.
Yeah.
Okay, and this is a technicality, but so when we're actually doing the blood draws, do we want to just draw 70, or do we have to draw 70 plus 20 for a total of 90?
Oh, 70 total. You know, we can't go over. So about 20 to Eliquid or store.
And then 50 for analysis.
Yeah, so that will be all determined by what, how much each company wants.
Understood.
Yeah. But basically 50 will go to each collaborating laboratory that will be doing ctDNA, proteomics, transcriptomics, you say various liquid biopsy laboratories, and then 20 will be stored for future research.
Okay, so do you want to change the language in the protocol? In the protocol, we said 60 ml, 60.
Let's change to 70 total.
70 total. So we'll say it won't exceed 70 mLs during one study blood draw.
Yeah.
Okay. Then 3 months. Okay, I think that's it for the consent form then. I'll make the necessary changes and then send it out to you guys, and then I'll start looking into the IRB process. Yeah, and then so that's that. Just a couple things related to pacemakers. So, Eileen and Sally. Eileen is done with her video editing. She's just awaiting the thumbnails, and she's good to upload. Sally is almost done with Paul Sean's. Also waiting for the thumbnails, and that one she's splitting into 3 videos because the clip is really long.
Yeah.
And then we conducted one moment. So we already did Madeline. Eileen did that. She's working on that as the next kind of round for her. And so Sally is supposed to do Marissa this Friday, but she is currently COVID positive. And
so if she's 5 days out and she becomes asymptomatic, as long as she wears mask, that's our hospital policy. If you quarantine for 5 days and you're not symptomatic,
yeah. So
I don't know, by Friday,
yeah. So she's still symptomatic. Hopefully she will test negative and be asymptomatic before then. But yeah, so we kind of put her scenario in the CDC kind of quarantine tool, and according to CDC, she can leave the house, but she needs to wear a mask until the 7th. But then the school guidelines have it such that she has to quarantine until the 7th, unless she tests negative before then. So she's going to be testing.
Go by the CDC, but testing positive, negative doesn't matter. Like some patients are asymptomatic and still positive for a couple months.
So I just wanted to make sure you're comfortable having Sally still conduct the interview.
So if she becomes totally asymptomatic, because she's quarantined more than 5 days, I am fine with her interviewing.
Okay. So then most likely, I think Sally will either test negative before Friday or become asymptomatic by then. So I think we should be okay there then. And then just one more thing. So Rachel was supposed to interview Lois. And she's reached out to Lois multiple times now and hasn't heard back. So, Rachel's emailed her, she's texted her. For some reason, she's been having difficulty getting in touch with Lois. So I was wondering if you knew of any better ways to contacting her or what you wanted to do.
Lois Harris. I just saw her yesterday in clinic. So you have to show up on clinic day and meet her and interview there. That's your best bet, because she's very weak. Yeah.
Okay.
So look at my schedule and figure out when she's coming. She's pretty much coming weekly because she's very weak.
I see.
Yeah.
Okay. We'll coordinate that with Rachel then.
Okay. And I think the thumbnail, we can't only rely on one person, Shana. So maybe whatever she's doing, we can make it like standard procedure, like this, and maybe learn from her. And so anyone who makes the clip can still work on it. Like I still want Shana to help and work on it, but I think her, she has family emergency. She's not able to work on it, which is become the bottleneck of everything. So I think we should document and kind of standardize everything so that people can still do it. I mean, it's not difficult. So
I'll double check with Shana.
Yeah. All right. Anything else?
I think that's it.
Okay.
We'll see you tomorrow in the lab, doctor.
So tomorrow is Wednesday.
Yeah.
So I will send you the link to the Tuma board so you guys can all listen in. I will be going into Tuma board remotely. I don't think I'm going to come into the lab tomorrow. So just come by 7:30 and then go into the link directly. And then you can listen to the Tumo board. I think Eugino, you have a link. Do you have that link or me? I don't know.
You sent a link when you presented Albert Curie case last year.
That's the same link. That's the same
Okay.
link.
Gotcha.
So just go to that link, share that with the group. At 7:30, just come in and then go and listen. That's 7:30. So we'll do the book club just like same thing. 7 o'clock, 6. We'll meet through Zoom 6 o'clock.